published enormously in this area, and he has been collaborating with ICGB for a long time on many other my colleagues, immunologist colleagues. His recent interest has been on therapeutic
Recording in progress.
His recent interest has been on therapeutic antibodies and their usage, and how to identify these therapeutic antibodies from the library by screening against various pathogens as well as against various proteins which are expressed by the pathogens. So I invite Dinker to tell some of his work. I must tell you that Dinker still has been really involved in research as much as he is involved in various official matters in handling ICGB affairs. So that's why we have been very much impressed by this dual role at ICGB. Now I invite Dinker to present his work.
Okay, thank you Pawan for this nice introduction. I have given a topic which in contemporary sense is not necessarily very fashionable, because people presently have shifted their focus towards COVID-19 in various levels. But even to understand COVID, the kind of things that I'm going to talk about would actually be not completely misplaced. So what my lab has been working on for a long time is with regard to antibody cross-reactivity, which we initially interpreted as molecular mimicry, and then moved on to understand immunology associated with it, and then there were conflicting interpretations of observations that we discovered, and we developed a lot of skills along the way. So on one hand I will tell you about how we evolved, and on the other hand how we will go further. So that is part of the process. So quite a few years ago we actually were making hybridomas against carbohydrate moiety and checking those antibodies to be cross-reacting with a peptide molecule, and that had a background which I am not preparing, but suffice to say that we had antibodies which were very specific to monocyte moiety, and also there were antibodies in that mixture of population of hybridomas which cross-reacted with a peptide molecule. And we picked up those which showed high signal in ELISA, and we could see even in the measurement of affinity, we could see there would be clearly antibodies that would recognize only one moiety, and there were antibodies that could recognize both the moieties with equal affinity. So it was very clear the polyclonal population when you immunize animals would actually have both types of antibodies, those which can, and this is, I am talking about monoclonal. I am not, I completely skipped the polyclonal studies, but monoclonal antibodies you could make specifically which have very high level of specificity, and you could actually identify in that population antibodies that cross-react with something completely unrelated. And that unrelated moiety, which was a peptide as against a sugar moiety, we originally began thinking these two molecules may have a similar topology. And we did lot of studies along the way to try and understand their structures, and I have here three-dimensional structure of antibody complex with the peptide as well as with the sugar moiety. And it was very clear both bind at the same site, but the interaction involved in binding to the antibody molecule were very different. Their topologies were very different. So you could actually see cross-reactivity of antibodies with no similarity between the two molecules, and that became a starting point for us to actually understand why cross-reactivity actually occurs and what is the significance of it. So this was established almost 8 years ago. Then we discovered subsequently many structures in the literature reported. You do see cross-reacting antibodies. So you do have antibody against a couple of slightly different viruses showing cross-reactivity. You also have one of the very well-known growth hormone with cross-reacting with another growth hormone. And actually one good example of this cross-reactivity is anti-idiopathic antibodies that have been reported. In some sense, that also becomes a similar kind of situation. So the question is, why is immune system making polyreactive antibodies when it is generally believed that antibodies have to have a very high level of specificity, and many diagnostics for diseases have been developed on the basis of that hypothesis. Now we actually realized, after understanding sufficient amount of immunology, that of course there has to be certain amount of specificity. You do have antibodies that can have the ability to discriminate subtle differences to ensure specificity and fidelity of the immune response, but there will be antibodies which so there is presumably there is another set of antibody clones which possess natural plasticity, and probably they are designed to disregard antigenic variations. In a way, nature or immune system has found, in our point of view, a solution to the problem of immune escape is what we thought. And this was a good example of that we probably were able to discover. So that leads to a dichotomy. On one hand, you have presence of polyclonal antibodies that show cross-reactivity or polyspecificity, and they are available. And if you look at the experiment that we did in mouse model, we had almost half of the hybridomas that we made were of cross-reacting type, and the other half were highly specific to the epitope against which these antibodies were made. But then we do have a situation that we have been often been told that immune escape is something that is common to HIV and influenza virus. So if you have cross-reacting antibodies, there should be antibodies available there, even if there is a mutation designed for immune escape, that should actually take care of it, and antibodies should be able to recognize any mutant strain is what we thought. And so we decided that we will actually ask this question systematically and address it more specifically in a physiological context to understand how does polyreactivity indeed exist for the purpose we think it exists, and we analyze So the good examples of this will be HIV and influenza virus, and we have actually been working on epitopes from both influenza as well as HIV. I don't need to introduce influenza virus for the people participating in this, but I will just say that antigenic variations allow the pathogen to escape from immune system. This has been established. If I actually say, yeah, but polyclonal poly-specific antibodies exist and it will not happen, nobody will accept our publication. So this is something that has been accepted as a gospel truth. So our question was in terms of what is the relevance of the polyclonal antibodies that we were seeing. And so we decided to take well-studied epitope from a surface protein, which is a hemagglutinin, from influenza virus, and for which both structural and immunological understanding has been very narrow. And the epitope that I am talking about is highlighted in red here. And we also picked up from the huge library that is available of natural mutations that take place in hemagglutinin molecule of influenza H3 and 2, and picked up the most abundant of these mutations specifically in the epitope that we identified, and 8 of those we actually synthesized. And what we did is, the idea was to make monoclonal antibody that is specific to that particular epitope. Not to take the whole antigen and make antibodies, because then they will be all over, and picking up antibody which is precisely defined for a given epitope would be not easy to identify. So we took the approach to accept the epitope as the logical choice for making antibodies, and then decided to check the cross-reactivity with the natural variants of that, and those are about 7 of those here. And then we adopted the fast display library technology. Again, I will not go into the details of this, for which a few years back there was a Nobel Prize given for developing this technology, and so we had this library procured and adopted in the lab, and then we screen against this peptide epitope WTGVTQN, and what you see in the red dots here are the basically cross-reacting, which is almost negligible in this case, and we actually screen in case of this epitope about 2000. What we have listed here is 1600 phages, and so a large number of them showed very high antibody binding, and so you can see a pool of diverse phage clones with varying affinities from the monoclonal phages, and about 100 of these we first picked up to check the polyreactivity, and we found they are actually, most of them show cross-reactivity. What we have shown here in the red dots are those which are highly cross-reactive, but if you look at the total population, you would notice one mutant out of this 7 mutation from this, particularly from tryptophan to arginine, majority seems to be on that high cross-reactivity level. This is still at the phage level. So, but we nevertheless picked up all those high binders and then checked the cross-reactivity by ELISA, and then you can see most of those show cross-reactivity. Even among the high binders, you will see on the lower end is that W2R mutation only. We then cloned about 12 of these high affinity binders and made sequence those clones, and then actually used that sequence data to make single chain antibodies from this. And this is just to show sequence, but we have purified those antibodies subsequently and made it in large quantities, and we can see even at the antibody level, you can see the binding there, and we have checked the binding affinities by BLI as well as Biacore, and they are between 10 raised to power 6 and 10 raised to power 7. Some of those were at the level of 10 raised to power minus 8 also. But what is important is that we were able to make about 12 antibodies which are showing cross-reactivity with all the 8 variations of the influenza virus, hemagglutinin, and not only that, the affinities are nearly in the physiological range. We actually kept our cutoff to be about 10 raised to power minus 6, but we could try and be more stringent as well. And what we actually discovered is all of them, even phage of course we had demonstrated, but even at antibody level we are showing the cross-reactivity. We made hemagglutinin expressed on the cell surface, and even the whole hemagglutinin expressed on the cell surface actually could show binding. So in that sense, in this cell surface environment, which would be almost analogous to hemagglutinin on the viral surface, these antibodies bind. So in that sense, antibodies made against a peptide epitope can bind to that epitope when it is in the protein environment also. So I'm jumping the story just to get it done quickly. So having made these antibodies, we purified. We earlier attempted crystallography of these antibodies with the complex so that we can understand the structure, but we were for quite some time we were not getting these antibodies crystallized. Only in last year or so, all my students are now able to make crystals of antibodies as well as their complexes with the antigen in the lab crystallized and looking at the structures. But at that time, we took advantage of Schrodinger availability, and we made structural models for all of those. And one important observation that we got from the early data, which was when we measured the affinity of binding between the antigen and antibody, what actually became evident is, of course, the affinity of binding of the antigen and antibody were comparable. I actually could have given it for the number, but I am showing this picture only to highlight the second mutation, that is W2R. So even in the models that we have made, the mutation which corresponds to from tryptophan to arginine shows actually poor affinity. So in that sense, what we found from the phages in large population, antibodies that are made against WTGVTQN seem to not cross-react with RTGVTQN. So that seems to be such that even if you have a cross-reactivity, there are certain mutations that may not actually be amenable to this cross-reactivity. And that was the key data in terms of what we were able to interpret. So one thing is very clear from, if you look at the data that has been reported in place of influenza virus, hemagglutinin, or the infectivity as a whole, you have, if you actually calculate what are the possible number of mutations or rate of mutation, if you take that into account and each mutation is responsible for bringing you immune escape as established in the literature, then the influenza epidemic should happen much faster than what you actually see. And this has been analyzed actually to show the actual total number of mutations in the hemagglutinin that are possible from the natural mutation data in the database. Not all of them are actually leading to immune escape, but it's only a small proportion of that. So cross-reactivity exists even in case of influenza hemagglutinin mutations, but not all mutants are amenable to cross-reactivity. And that seems to be the key in terms of the dichotomy that we raised, that you do have mutations which can lead to immune escape, and there are mutations which actually overcome that. And this population is probably 1/5 of total mutations that seems to be very critical hotspots in that case. And that now whatever is there in the literature seems to be consistent with the experiment that we carried out based on our studies. So what it actually could mean is antibody promiscuity could explain limited diversity and discontinuous antigenic change under the effect of antigenic trait. So having established that, in the process we had also established the ability to make monoclonal antibodies single chain in our lab. So excited with collaborations available within ICGB, particularly in the area of malaria and tuberculosis. We realized we should be able to begin using this technology, and that is part of the study with multi-partner collaboration. So we decided to start making now therapeutic antibodies in various cases. Rather than asking fundamental questions with regard to immunology, we decided to actually bring it to the practical use. I will not dwell upon this slide because it will only tell you what you know, but you have various combinations of designs that are available for making therapeutic antibodies, and one which we are using is the fourth one in this list is single chain antibodies. So it has been used, and there are some successful therapeutic antibodies which are single chain GFP region. So we decided to target, we are of course working with Pawan Malhotra on malaria antigens, but we set out to target with whatever processes we had established in the lab to look at the surface proteins of Mycobacterium tuberculosis, and we have picked up 3 proteins, out of which for 2 of those we have been able to successfully make monoclonal antibodies. And the first target protein is antigen 85 complex, which has 3 different proteins, 85A, B, and C. And the crystal structure of antigen 85 complex is also known, and the region which is important functionally for this is also defined. So we picked up that region as an epitope, and then went out to search in the fast displayed library for antibodies, and we have taken into account various considerations in terms of making these antibodies. We'll come to that later. So we screened, as we did in case of influenza virus hemagglutinin, large number of antibodies, checked those for specificity, and also checked for cross-reactivity. You can see there in step 4, you do have many antibodies phages there which can cross-react with A, B, C. Because if you have antibody inhibiting only 85A, it will still it will not have any effect on the pathogen, because B and C actually carry out the same function. So you need to have the antibody that can bind to A, B, C both, and we could actually check out to ensure that we pick up phages which will be of that type. And we picked up those phages, we purified the antibodies, and we were able to make about 10 antibodies, out of which 9 of them were cross-reacting antibodies to antigen A, antigen B, as well as antigen C. And one of them was highly specific to antigen A. So one thing is very clear, that we have antibodies actually that behave more or less similar to what we were expecting in our mouse model studies. We do have a mixture of cross-reacting and non-cross-reacting antibodies even in the phage library, and we have cross-reactivity. We have also been able to crystallize it, and what we are now trying to do in collaboration with Dheeraj is to try and work out functional assays to see whether these antibodies will work towards being able to the function of antigen A can be inhibited by these antibodies, and go on further. So we have another antigen called LPQH, which is also a surface protein. It's a lipoprotein, and we did the same thing. We picked up this peptide, identified monoclonal antibodies, and picked up at least one antibody, which is single chain antibody ZH288QH, which binds not only to the peptide against which it was raised, but almost comparable affinity. It can also bind to the whole protein which was expressed and cloned in the lab, and in the antigen 85 also is being expressed to check it with the whole molecule and continue further. So these are the two antigen against which we have antibodies in the range of 10 to the power -7 or 10 to the power -8, and we should be able to check whether they are in a position to inhibit. Then of course after that we will have to go on to see if we can, how we can deliver it, and see if in an animal model they would work. So to summarize, what I talked to you about is to show significance of antibody promiscuity in physiological situation, and we are convinced that it's natural for immune response to be cross-reactive, and we could identify strongly cross-reacting antibodies in case of influenza virus epitope variants, and majority of epitopes of course could not escape immune surveillance, but there will be some mutations which will escape, and that was evident even in our data. The mutations beneficial for the virus of course will be more important there, and delay in the appearance of new antigenic variants may be contributing, is being contributed to antibody promiscuity, in the sense that if you look at all these combinations and permutations that are possible for making mutations, if all mutations were escape mutations, the frequency with which we'll get influenza will be much higher. But appearing the epidemics were much less frequently, and that is because we have escape only among the very few hotspots, and not every mutation is an escape mutation. And we have used this technology further towards developing targeted specific antibodies against tuberculosis surface antigens, and this is the work that is ongoing. And the TB work is being carried out by Stabonia and Zaid presently, and mainly Sheenam was the one who carried out the influenza work, and almost everybody in my lab in last 15 years have actually contributed to this research, and thank you for your attention.
Hello.
Thank you, Dinkar. Yeah, I must tell you that we have also been lucky that we have been able to get antibodies, single chain antibodies against 3 malaria parasite protein. And we have some question from the chat box.
Presentation.
Akash Pratap from CSAR IGIB. I have a question that can we say that response charge on the molecules because the antigen is protein, then more immune response generation, while sugar moieties and lipids generate less immunogenicity because less charge on it.
I would not say that. We have actually studied many monoclonal antibodies in our lab. We have Akash Pratap, we have
Yes, sir. Yes.
question.
Yes, sir. Yes, sir.
The
Hello, sir.
answer to that is I won't link it to chart because we have many monoclonal antibodies studied in our lab which actually bind to hydrophobic surfaces.
Sir, sound is not clear.
Can you hear me now?
Yes, sir. Yes, sir.
We have come across, we have raised antibodies against hydrophobic molecules also. What probably the question that you may want to ask is the antibodies against the charged amino acids being more cross-reactive. The answer to that also is no, because we have analyzed sequences of epitopes in many cases, and we have not been able to link any amino acid, specific amino acid properties to the nature of antibodies, whether it is flexible or not flexible, whether it is cross-reactive or not cross-reactive. So it is more to do with topological features of the epitope that matters in terms of antibody response. Thank you.
Okay, sir. Okay, sir. Thank you, sir.
Thanks, Dinkar. I think you can post more questions for Dinkar on chat box. We will certainly send you the answer back. Because of shortage of time, we now move to the next talk by Professor Gavin Wright. Hi again.
Hello, I'm here. I'm trying to share my screen, but here we go. Right.
Yeah.
Can you put it on full display?
Yeah. There we go. Is that clear now?
Can you move your slides forward so that we can see if it's working?
Yeah.
Yeah.
It's working.
Okay.
Okay. Okay. Our next speaker is Gavin Wright, professor from York University, and I thanks Gavin for accepting our invitation immediately. Gavin has been doing really fine work on the parasite surface protein and host parasite interaction. He has developed many novel technologies to his credit, and his main aim is actually again to get to develop surface molecules, and he has been instrumental in understanding various novel surface host receptor interactions. So, and I give stage to the Gavin for his presentation.
Okay, thank you very much indeed for this invitation to present some of the work that we've been doing recently in my laboratory. It's a great shame that I can't be there in person. I really enjoyed my last visit to the Ccmb, and it's certainly a nice place to go when the weather's so bad here in the United Kingdom. Anyway, so what I'm going to talk to you about today is a disease of livestock, and I'm going to start with this photo here, an all too typical scene from a village in sub-Saharan Africa. And this man here, you can see, is showing a great deal of concern for the health of this animal here. And he's looking really concerned because he knows that there's not much that can now be done to save the life of this animal. And this animal provides both him and his family with food, milk, and draft power. It's very likely that this animal also represents a significant proportion of his personal wealth. And the disease that this animal is suffering from is called animal African trypanosomiasis. And this is a parasitic disease caused by parasites of the genus Trypanosoma. And there are two species of this parasite that cause this disease primarily. And they are Trypanosoma congolense, and the one that we're going to be dealing with mainly in this talk today is Trypanosoma vivax. They're both transmitted by tsetse flies and live in the blood of the host. You may be more familiar with a different species of trypanosome, Trypanosoma brucei, which can infect humans, and that causes sleeping sickness. These parasites infect very important livestock animals, primarily cattle, but also goats, sheep, and pigs. And the consequence of this is it makes vast areas of ranching land in sub-Saharan Africa completely unusable, creating what's known sometimes as a green desert. The impact of this disease is absolutely vast. So 3 million cattle will die from this disease annually, leading to a direct economic impact estimated at several hundreds of millions of dollars. Such is the impact of this disease that the Food and Agricultural Organization of the United Nations stated that this disease lies at the heart of poverty in Africa. The disease is currently managed by drugs, although resistance is now widely reported and is increasing. These parasites also infect wildlife, so there's a wildlife reservoir. So this would, of course, make eradication using drugs impractical. And as with many parasitic disease, there's no vaccine available. So how do these trypanosome parasites thrive in the blood of their hosts? Well, perhaps the most famous mechanism that is used is one that's known as Antigenic Variation. And this depends on the serial expression of an allelically excluded and very abundant cell surface glycoprotein called the variable surface glycoprotein. And you've probably studied this in lectures. So this is what happens typically is if this is parasitemia as a function of time, upon infection, the parasite is able to multiply in the host, which is actually then able to generate a quite effective antibody response, which is able to control that infection. But within that population, you get switching to a different antigenic variant of this VSG, which then results in a different population of parasites expressing this different antigenic variant of VSG, which then dominates the infection. So whilst individual parasites die, the infection as a whole continues. And what happens is you end up with these kind of classical waves of parasitemia over time. Another way that trypanosomes protect themselves from host antibody responses is that they can rapidly internalize them through a structure known as the flagella pocket. So this is an image of a trypanosome. It's a single-celled organism. It has a flagellum here, which lies along the surface of this parasite cell body. and there's a structure here known as the flagella pocket. And what you're looking at here are fluorescently labeled antibodies in green, which are very rapidly, and you can just see from this very nice time course here, that within a minute, these antibody complexes are internalized inside the parasite and degraded. And so the parasite is able to protect itself from host antibodies using this rapid endocytosis. There are also more sophisticated approaches that the parasite can modulate the host immune response. So there have been some attempts to try and vaccinate against trypanosome infections. So the kind of classical approaches of vaccinating with inactivated or attenuated parasites has really resulted in only partial protection, so delaying the parasitemia. But of course, modern vaccines now are more chemically defined and known as subunit vaccines, which makes manufacturing more consistent. So there have been some previous attempts to use purified components to elicit protection. Those have been mainly done in Trypanosoma brucei, and there have been some work on these invariant surface proteins ISG65 and 75, as well as some results with cytoplasmic components, tubulin and actin, but neither gave very strong protection. So the collective of the experiments has been really that vaccinating against trypanosomes has not really been considered a high value option. But we posed ourselves the question using our protein expression technologies, which was to whether we could identify an invariant trypanosome surface protein that could possibly elicit protective immune responses. And the approach we took was using a kind of genome-led vaccine target identification. This is sometimes known as reverse vaccinology. And it works as follows, which is to start with the genome sequence of the parasite, and then look within the genome to identify likely vaccine candidates. So those are things that would be accessible ideally to antibodies, so cell surface and secreted proteins in this instance. What we then do in my laboratory is to express the entire ectodomains of these parasite cell surface proteins as soluble recombinant molecules. And we do this in a mammalian cell expression system with the idea that what we are going to do is improve or increase the chances that these ectodomains will be natively folded because they contain structurally critical post-translational modifications such as disulfide bonds, and they're more likely to be added if we express them in a eukaryotic expression system. And then the idea is to then produce a panel of these parasite surface proteins and vaccinate animals with, and then use an animal infection model to test to see whether we've elicited protection. And what we're using here in this murine infection model is a bioluminescent form of the Trypanosoma vivax parasite. So these are the selection criteria that we imposed on in choosing our vaccine candidates. So we essentially just had these three rather loose categories. So we preferentially wanted to select large proteins, those that were over 300 amino acids in the extracellular region. And we selected architectures of surface proteins that we knew we could express as biochemically active forms. So this would be type 1 cell surface proteins, GPI-anchored surface proteins, secreted proteins. And the idea of choosing these larger proteins is that they would project beyond this variable surface glycoprotein coat, which is thought to act as a kind of spheric shield against host antibodies. If you look in parasite genomes, you find very often that these proteins belong to large paralogous groupings, and we were concerned that there may be some functional redundancy there. We didn't want to target one protein only to find that its function was substituted by another antigenic different protein. So we selected those proteins that did not belong to these large paralogous groupings. And of course, we wanted to choose those proteins that were expressed in the bloodstream form of these parasites. So using these rather loose criteria, we selected 60 proteins, of which 39 we could express at sufficient levels to purify and vaccinate animals. And this is a protein gel of those candidates. I've segregated them into these different functional architectural classes, so the transmembrane proteins, GPI-anchored proteins and secreted proteins, and then they've all been given this rather prosaic numbering system, so V and then just this number here, which helps us in the laboratory, writing them on tubes and so forth. This just gives a little bit more detail about the infection model that we are using. It's a transgenic Trypanosoma vivax parasite that's expressing the that expresses the luciferase gene, which allows us to use bioluminescent imaging to quantify parasitemia longitudinally. And these are the types of images that we can get. So at day 2 of infection, day 5, and day 7, you can see from the emission of this light from the luciferase gene in the parasite that we can quantify in a longitudinal fashion the amount of parasites in the animals. And this way of measuring parasitemia we found to be more sensitive and more reproducible than, more consistent than doing direct counting of parasites in the blood by microscopy. And this is the same infection here, quantified by microscopy and bioluminescence. You can see that we can detect the infection much earlier using these bioluminescence readings, even at day 2 post-infection. And the data points are often more consistent. It's an acute infection, so we have to stop the experiment in animals at day 9 typically, when we use a low dose of parasites, so that would be 10 to the 2 parasites in the inoculum, but it's very reproducible. And we maintain the parasite by serial passage in donor animals. That's because you can't culture the bloodstream forms of this parasite in vitro. This is the vaccination protocol that we use. So it's a prime followed by 2 boost model. We're using a protein in adjuvant formulation. We're using alum as our adjuvant. We take blood biopsies just after the final boost to quantify the elicited antibody titer. Importantly, we include a rest period here from the final boost before we challenge with a parasite given from a donor animal. That's to avoid any kind of general systemic immune stimulatory effects that might be elicited by the adjuvant, and then we monitor the infection using this bioluminescent imaging. We found that when we took the parasite out of a donor animal, it's actually quite labile, and you gradually lose infection if it's outside of the animal for too long. And so in order to avoid being confounded by these results, what we did was to have this kind of experimental setup here, where we had a cage of control animals that, which were infected first, then the 4 query proteins in a cohort of experiments, and then a second control of control animals which were infected last. And only if these 2 controls agreed with each other, having the same kind of infection kinetics, did we trust the data in here. This just shows you the quantification of the antibody titers elicited to each of these different vaccine candidates. There's not too much to take from this slide, apart from to say that the antibody titers between proteins varied, but generally were reasonably high, having a half maximal antibody titer in the kind of tens of thousands-fold dilution range. So this slide represents the results of this vaccine screen that we did. It looks like a busy slide, but it's quite simple to understand, and I like to show all of the data. So this took quite a long time to generate. So what you're looking at here on the y-axis is this is the bioluminescence. So this is a proxy for the parasitemia as a function of time. So they're done in these cohorts. And so here you can see in blue, these are the control animal infections. And you can see that we get this rapid and controlled and very consistent infection with these animals. Where we've got these V numbers here, these are the different vaccine candidates, and you can see that most of them, which are highlighted, indicated in black, don't actually affect the infection at all. Where I've highlighted in this orange color, these are candidates that statistically did have an effect on the parasite infection. Sometimes that was quite mild, and in other cases was reasonably good. It delayed the infection a bit. But we have one candidate here in cohort 8, this V23 protein, which was able to prevent all parasitic growth. So these are process data. I like to show the underlying images. So this will be the kind of level of effect that we have with, let's say, this V8 candidate. You can see by day 7, you're seeing a reduction in the amount of parasitemia there. This is the V31 candidate. This was perhaps the best of these other 4 types of delaying candidate here. So you get this reduction in light production. And in the V23 immunized animals, we were unable to detect any parasites. So we repeated this with larger cohorts of animals. The V23 candidate repeated very nicely. This is now a cohort of, this is now a group of 15 animals. From day 6, 7, 8, and 9, there were no detectable parasites there. This is the V31 candidate. This actually performed better in our repeat. But again, you can see that there is some parasitemia gradually developing, but still it is able to control infection to some degree. The 3 other candidates, the 2, 8, and 59, they did not replicate in these larger animal studies. So what is this V23 protein? So it's a typical type 1 hypothetical protein. It's not been annotated at all in the genome. It's a glycoprotein. There are 8 N-link glycosylation sites predicted in its extracellular region. You can see you get these smears here on when we purify the protein in the gel. These likely represent glycoforms. This is the accession number here. It has a very short cytoplasmic region of just 18 amino acids. There are no close paralogs to any protein in the Trypanosoma vivax genome, nor are there close orthologs in other Trypanosoma parasite species. So, as I mentioned, you can't culture this parasite in vitro, at least the blood stages, and so that makes doing any kind of functional studies very difficult. So to try and understand something about the functional role of this protein, we turned to just trying to identify its location on the parasite. And we found that it is located here, just at the boundary between where the flagellum lies alongside the surface of the parasite cell body. So this is just an EM image here of a trypanosome. This is the flagellum here that lies along the surface of the body here. This is what it uses for motility. The flagellum emerges from this structure known as the flagellar pocket and lies then along the surface of the cell body. And it's here, at this nexus between the two, the flagellum and the cell body, that this protein is concentrated here. This is using antibodies and immunocold labeling using electron microscopy. So we wanted to understand a little bit more about the mechanisms of immunity, and one obvious question is, do antibodies play a role? And the classical way of doing this is through passive transfer experiments. So we took some pooled sera from vaccinated mice, and this is the isotype balance within that pooled sera. So it's dominated by IgG1 isotype antibodies. And then what we do is we dose animals the day before, on the day, and the day after, being challenged with Trypanosoma vivax parasites, and then follow the infection. Using this passive transfer experiment, you don't get quite the same level of antibody titer as we would with vaccination. But still, we were able to show a dose-dependent inhibition of parasite growth using these through this passive transfer experiment. So again, this is bioluminescence, this is parasitemia here, and these are the V23 immune sera relative to naive serum control. So you get this dose-dependent inhibition of parasite growth. We wanted to explore this in more detail, so we made some monoclonal antibodies. These are all of the IgG1 isotype, and using a dose of 100 micrograms in the same three doses protocol, we could find that 3 of these antibodies were able to passively protect against infection, whereas these 3 antibodies were not able to protect relative to this control here. So that was, 3 out of the 6 were able to confer protection. We could not correlate any property of the antibody to its protective efficacy. And we measured binding affinity of the antibodies to the antigen, and also we could crudely map roughly where the epitope lay, which we crudely categorize as in the N-terminal region, the middle part of the protein, or the C-terminal region. So this is the relative protective efficacy from from the last slide, so the 3D12 antibody here you can see is the most protective. This bound the middle part of the protein. These other protective antibodies bound the N-terminal region, but we do have an N-terminal region binding monoclonal antibody that is not protective, this 8C9, which actually is high affinity. So these are surface plasmon resonance traces looking at the binding affinity of the antibodies. So this is binding as a function of time. You can see these antibodies are very high affinities. The interaction doesn't dissociate over the time course of the experiment. This is actually our lowest affinity antibody, this 8E12 antibody. You can see a nice dissociation here. That was actually quite a protective antibody. So this suggests actually that perhaps these antibodies are disrupting some kind of protein interaction on the surface of the parasite. Now, I stressed the fact that all of these antibodies were of the IgG1 subclass, and in mice, this particular isotype of antibody is not effective at recruiting immune effectors, such as it's not very good at fixing complement, nor does it bind to activating Fc receptors. Whereas other isotypes, such as IgG2 isotypes, they're good at recruiting complement and binding to activating Fc receptors. So one experiment that we wanted to try was to switch the isotype of the 8E12 antibody from an IgG1 to an IgG2A isotype. And we could do this using a method that we developed quite a while ago now, which allows us to express on a single plasmid monoclonal antibodies in a recombinant way. And what we did was to simply switch the heavy chain here from an IgG1 to an IgG2A mouse isotype. This is again just showing you through using some surface plasmon resonance traces that as a result of this isotype switching, we haven't affected the antibody specificity or affinity. And when we use this IgG2A formatted monoclonal antibody in passive transfer experiments, we got this really remarkable result, which was that they were extremely protective. So this is looking at a 50 microgram dose here. This is parasitemia. This is the IgG1 isotype antibody. This is the data point I've shown you before, showing you that partial protective effect here. But then when we switch to this IgG2A isotype, we get this very potent protection here. And we can make a dose titration of that, and we could find that just 20 micrograms of this monoclonal antibody was protective, which is really very potent indeed. Now, because we've expressed the antibody recombinantly, and it's an IgG2A, what we can do is actually mutate specific sites on the antibody that recruit different immune effectors. And that then would allow us to pick apart exactly what the contributions are to the mechanisms of immunity here. So I'm going to show you that here. So again, we've got bioluminescence here. So this is parasitemia on day 6. This is the data point I've shown you previously, showing you the IgG2A potent protection. And then if we mutate out the complement recruiting C1q binding site on the antibody, we relieve that inhibition almost back to the control antibody, suggesting that complement is a very important mechanism of protection in this particular setting. If we mutate out the Fc receptor binding site, again, we get an inhibition of that protection, but not quite to the same degree. And if we mutate out both of those binding sites, we basically convert our IgG2A monoclonal antibody to the same potency pretty much as an IgG1 antibody, which is just a little bit more, which shows some protection relative to this control here. So this tells us that there are probably 3 protective mechanisms going on, the most important of which is complement recruitment, the second, the ability to bind Fc receptors, and thirdly, just the direct opsonization of the protein with the antibody has an effect also. It's known that many parasite vaccines can fail due to strain-specific immunity, due to polymorphisms in the protein. Collaborating with Andrew Jackson from the University of Liverpool, we could show that this V23 protein actually was really quite well conserved across 29 isolates that we had from different geographical regions of this particular parasite. So it's actually really quite a well conserved protein. In fact, the only mutation that we could find, the non-synonymous mutation, was just here, just prior to this is the transmembrane region here, so it's just here in the extracellular region. We could show that V23 is not immunogenic in the context of a normal infection. So this is an ELISA. So this is the ELISA reading here. What we're looking at are the antibody responses to different Trypanosoma vivax surface antigens. So this is that V2 protein, V53, and our vaccine candidate, V23. And these are cattle who have got diagnosed infections of Trypanosoma vivax from both Cameroon and Kenya. These are the controlled UK cattle. They don't obviously have any response. They've seen the parasite. The Cameroonian and Kenyan cattle do show a response to these antigens, but do not show a response to this V23 protein, suggesting it's somehow immunologically protected in a normal infection. And then finally, we just needed to show that we could, this protection was long-lasting. And so what we did was to take our vaccinated animals and those that were protected, we challenged them subsequent times. So this is the first challenge here. You can see that the control animals get this rapid, uncontrolled parasitemia. This is the protection of the vaccinated animals, which we then challenge again about 3 months later. The control animals again develop good infections. The vaccinated animals are then protected. And we can go right out today at 130, showing that this protection is long-lasting. So I'll conclude there and say that we've identified an invariant vaccine antigen, which we call V23, which is capable of eliciting sterile protection in a murine model of Trypanosoma vivax infection. The antigens are localized to the parasite flagellum. I can show you that antibodies are important through these passive transfer experiments, and by dissecting out the immune recruitment sites on a gamma-2A antibody, we could show that complement was a major immune effector mechanism. And we're now trying to take these experiments through into some field trials. Finally, I'd just like to acknowledge the contribution of Delphine. She's a super postdoc in the lab, and she did the vast majority of this work, but she's been helped by many others along the way, which I've listed here on the slide. And again, thank you for your attention, and I'm happy to take any questions you may have.
Thanks, Kevin. Very nice. So what is the next plan? You are going to take it for the field trials? So Gavin, you would like to take this to the field trial now?
I'm happy to take questions.
Yeah. Gavin, it's Pawan from ICGV. Are you
Hi.
planning to take it to the field trial now?
Oh, have we done the field trials now? We're currently obtaining the funding in order to do that. So we're not there yet. It's going to be a 2 to 3 year kind of project to do all of that.
Okay. So there is a question from Shreya from NII, our neighboring institute, that is there any correlation with the antibody titer and antigen concentration in terms of protection?
A correlation, did
Yes.
you say, between yeah, no, not an obvious one that we can see. The main correlation is with the isotype of the response. So let me see if I can find the So these were the isotype balances when we used alum as an adjuvant, which is known to elicit in mice predominantly gamma-1 isotype. If we switch the isotype to something like QuIL-A, that will increase the ratio of IgG2A antibodies to IgG1. And as I've shown you, those are more potent in protection. And so actually, if we switch adjuvant to QuIL-A, we actually get better protection than if we use alum. So we haven't been able to get, there is a correlation in the sense, so I showed you this here, which is the passive transfer of the immune serum. This protection is not as strong as if you used vaccinated animals, and that's because when you passively transfer antibodies, you don't get quite the same titer. It's actually lower. So I think the answer is actually yes. So there is a correlation between the titer of antibodies as well as the qualitative nature of the antibody response. So if you have the IgG2A antibodies versus IgG1 in our mouse model, that certainly helps with protection. Yeah.
So Thanks once again, Gavin. It has been a wonderful talk, and I hope this molecule you will be able to take further for the field studies, and then maybe in the vaccine part it is in the market. Thank you very much. We are grateful
Thank you.
that you accepted our invitation. Thanks a lot. Vinay,
हाँ
full screen full screen से एक बार slide check कर लो, move करके.
चल रहा है.
चल रहा है? Good, good.
Is it working for you guys?
Yeah, yeah.
Okay.
So it's my pleasure to introduce Vinay, a good friend and now the director of Ccmb. Vinay has again done exemplary work in the area of Mycobacterium tuberculosis. I knew earlier Vinay has been doing work on various drug targets, particularly some of the kinases. But when I talked to Vinay about the talk, he said, "Now this time I am going to give you some work of theirs on drug resistance in Mycobacterium and the role of DNA repair enzymes." So, Vinay, over to you. Please present.
Thank you, Pawan.
Menu. Can you change the mode to use slideshow?
It is not? Is it not moving?
No, it is moving, but it is in the menu mode. Yeah. Next one. Yeah. Correct.
Now is it okay?
Yeah.
Okay. So, thank you, Pawan, for giving me the opportunity to share some of our recent work. Very different from what I generally present, all mechanistic stuff. So this is on evolution of drug resistance in tuberculosis. Let me start with my lab. I mean, everything that I talk about anywhere is done, is basically because of the work done by these people here. For today, I'm going to talk about the work done by one girl, Sabahi. And I have had many students, past lab members, and the current lab members are around 7 or 8 of them, split between 2 cities. Hopefully that will change in near future. Mycobacterium tuberculosis, a very brief introduction. I really don't want to spend too much. It's an ancient pathogen, used to be the major killer, at least till COVID came along. Emergence of multi-drug resistance and extremely drug resistance tuberculosis is a problem, and that continues to be so. India does have the highest burden of TB, and that's why it's a problem that we need to anyway work on. So when people, this is aerosolically transmitted, when people come in contact with the infected person, the aerosolic droplets are taken in by our body, and that is taken up by the innate immune response, which is then adaptive immune response comes into the play and basically kind of surrounds the infected innate immune system, and that is what eventually leads to something called a granuloma. So the granuloma is basically walled off structure where the pathogen is inside in the infected cells, and your immune system is responding by walling it off. So under these circumstances, it has particularly 3 different fates. Either it becomes either completely gets eliminated, like in calcified granuloma, what you see here, or you can get an active disease, which happens in about 10% cases, where now it starts spreading into the other parts of the lung and even extrapulmonary tuberculosis. And otherwise, it is contained in the same granuloma for a long while to come, that is called latent tuberculosis, which upon an opportune moment can actually become an active infection all over again. So my laboratory, as Pawan pointed out, works basically on serine and protein kinases, and that's what I've been doing for 17 years. We also work on peptidoglycan synthesis, regulation of cell division, regulation of transcription, and even on a bit on host-directed therapy. For today, I will be only concentrating on one of the aspects which I started working on around 5-6 years ago, which is on evolution of drug resistance and DNA repair. So when it comes to tuberculosis, there are approximately 4 different antibiotics are given as, not approximately, 4 different antibiotics are given as the first line TB drugs. They are isoniazid, rifampicin, ethambutol, and pyrazinamide. And in addition to that, there are second line anti-TB drugs are given in case you end up getting resistance to any of the fluoroquinolones and injectable drugs are given. So if you have resistance to one of the antibiotics, it's called monodrug resistance. And if you have resistance to 2 antibiotics, it will be MDR. And if you have resistance to 2 of the first line antibiotics and 1 of the second line antibiotics, it's called XDR. So these are the definitions by which they are kind of go. And you know, when all these things happen, currently bedaquiline and delamide are the ones which are essentially given. One of the things is that drug resistance in tuberculosis is an important topic, and there are multiple papers that are published from various groups. We were specifically interested in looking at a connection between the DNA repair mechanisms and drug resistance, and we started mining the literature. We found that there is some level of data on how the drug resistance is connected to some of the DNA repair genes. And so we kind of decided to expand our study and look into how, if first of all, if the drug resistance is connected to DNA repair, and if so, in what way you think we think it will be important for the evolution. So, just to give you statistics, drug susceptible cases are approximately 83%, and the remaining 17% are drug resistant, and out of them, 54% are MDR-TB, means they are resistant to 2 antibiotics. 30% of them are XDR-TB, and then this works out to be around 0.39 millions in MDR-TB cases. India has the highest burden of MDR-TB globally. It contributes to 27% of the MDR-TB. That was in 2019. So how do you get drug resistance? There are multiple ways. One, you take them, you modify the target. So if you modify the target at a genetic level so that the drug does not anymore bind to the target, but it is not, then it obviously is also used to drug resistance. And other thing is modify the drug in itself. So, or change the expression levels of drug metabolizing enzymes, or change the, pump the drug out, or do not allow the drug to come in. So there are essentially multiple ways drug resistance can be evolved in not only in tuberculosis, but in every other pathogen. So you can modify the target, you can have drug can be modified too. You just decrease the concentration, intracellular concentration, all kinds of 4 different mechanisms essentially. So, however, even despite all the known mechanisms, there is 10 to 40% of the clinical isolates. The drug resistance cannot be determined even today because the mechanisms are, people are finding out what are the other efflux forms that are involved, stuff like that, and what are the other mechanisms. So we started looking at what are the other mechanisms, and this work is completely done by this girl Samanas in collaboration with my PhD mentor Umesh Varshney and Dr. Paritosh Kumar from the U South Campus, Sidra and Priyadarshini NII, Yogendra Singh from DU, who's her PhD mentor and a long-time collaborator. What she has done is that she basically looked into the available data. There are 2,700 genomes that were already available. There's nothing new. We just dug the literature and pulled out this 2,773 genomes, divided them into, based on the characteristics given, into either susceptible, monodrug resistant, multi-drug resistant, and various other things. And then we divided them, we looked into what are these 2,773. Most of them were lineage 2, but we found these strains from all 4 lineages. lineage 1, 2, 3, and 4. We divided them into all kinds of, as I said, into drug susceptible, MDR, XDR. And when we did the genome-wide analysis, which is really, I'm cutting it short, basically to say that we got around 170 odd mutations, or we got around 170 targets, out of which, as expected, there were some well-known direct targets which are all in red. Like, for example, if you're looking at isoniazid, you expect INHA to be a target, CATG to be a target, and we found that. So these reds are the known targets which are essentially validates our analysis. So, and there are many other efflux pumps we got, and some of the, usual suspect and some kind of expected to be there kind of things, and validated by other studies we got. But interesting aspect is we got here 4 green dots, which are DNA repair genes. So anyway, we started looking for it in the first place, but nevertheless, we were happy to see there are around 4 DNA repair genes that are found to be mutated in the MDR strains alone, not found in the, when we do the same analysis for susceptible strains, sequences from the susceptible strain, we don't find them. So why do we think DNA repair gene may be contributing to drug resistance? So we had a hypothesis. We believe that in the absence of any particular selection, in the absence of any particular mutations in the DNA repair gene, the pathogen relies on the spontaneous mutations, and some of these spontaneous mutations would help in evolution of the drug resistance. But if you were to have a DNA repair gene mutant, what would happen is your spontaneous mutation rate, or the mutation rate, generally goes up, essentially helping in faster evolution of the drug resistance. This was our hypothesis, and that is why we feel that DNA repair gene mutations may be one of the starting points for this kind of an evolution of the drug resistance. So what did we find? We found 2 DNA repair genes, 2 DNA repair. It's nothing. 2 DNA repair genes. One is a base excision repair, second is a nucleotide excision repair pathway, MUT-Y and UVRP. There was a specific mutation that was identified by GWAS study. So how do we even know that this particular mutation that is identified by GWAS is even relevant? In the absence of relevance, it is meaningless to say, oh, this particular mutation is actually responsible for the evolution of the drug resistance. What is relevance for something like a DNA repair enzyme? We expect it to, the mutation should some way or other compromise the activity of the enzyme. To do this, we needed a gene replacement mutants of these repair genes. Our Spengmatis mutants were provided to us by Professor Varshney for both MUT-Y and EPRP. For TB, we have generated it using gene replacement constructs using the traditional methodology, where we replace the MUT-Y gene with the antibody marker. So what we do under these circumstances is, once you have a wild type strain and a mutant strain, obviously you have what you need to do is you need to perform what you call as a mutation rate analysis. What you do is you grow for a certain time and enumerate the CFUs in the absence of antibiotics and in the presence of antibiotics, and based on the data you get, you calculate the mutation rate. We generally expect mutation rate to be high. So what we have is a wild type, a mutant, and we wanted to do mutation rate analysis. So these kind of things are only relevant if you actually come back and complement the gene that has been deleted. So we complemented with the wild type, we complemented with the mutation that was found in the GWAS studies. Here is R262Q or A524V. And you can very clearly see that the mutation rate, obviously, when you don't have DNA repair enzyme, mutation rate is expected to be higher, and it's approximately 5-fold higher. What is important is that when we put back the MUT-Y, it comes back. You can very clearly see it coming back. However, when you put back a MUT-Y with the mutation that was identified in our GWAS studies, it does not come back. So this indicates that this particular mutation that we have identified in GWAS studies seems to be relevant, and it is inactivating the enzyme. Same thing was identified, same thing was even found out in the case of UVRB, which is a nucleotide excision repair pathway. And we went ahead and validated these results even in tuberculosis, where we generated a mutant, and the results were very similar to what we got in Mycobacterium Spengmatis. So with this, we asked a particular question: how do you know? Would these mutant, would the deleting a DNA repair enzyme, would it provide some kind of an condition or some kind of an advantage? We did it one round of infection, but we did not find a major difference. So we went into a particular ex vivo evolution kind of an experiment. What we did in this case is we infected the cells, peritoneal macrophages, with any of these strains. Wild type, mutant, mutant complemented with the wild type, mutant complemented with the mutant. And after that 72 hours of infection, we took out the tuberculosis. We grew them for, after 120 hours total, we grew them for a while in vitro, and again put it back for a second round of infection. To make sure that we get some kind of a phenotype, we did either in the presence of antibiotic or in the absence of antibiotic. Means 4 strains without antibiotic, 4 strains with one of the antibiotics or the other antibiotics. But we kept on doing the same selection. In the sense, if there is no antibiotic, there was no antibiotic for the whole selection. If there was an antibiotic, there was an antibiotic for throughout the selection. So this was the idea I used to see if we are evolving any level of drug resistance or survival benefits when you keep on doing this with the DNA repair gene mutant. So what did we get? In the absence of antibiotic also, we can very clearly see that the mutant DNA repair mutant RB delta 1Y actually showed higher levels of survival. And when you complement it back, it comes back down to nothing. And I mean, basically where it was in the wild type. But when you complement it with the mutant that we have identified in GWAS, we actually have a similar impact as the deletion of this particular DNA repair gene, indicating that these two, this is compromising the functionality, and that does help somehow. Deleting a DNA repair gene helps in survival, better survival. Okay, and then we have done it in terms of antibiotic isoniazid, and in the presence of antibiotic rifampicin and ciprofloxacin. So the difference was more drastic when the antibiotics are stronger. Like in case of rifampicin, these antibiotics are relatively more stronger antibiotics compared to isoniazid, and the difference was almost 10-fold, 20-fold here. So, this was basically told us that not having a DNA repair gene helps in evolution of whatever it is, the stress, whatever the stress you impose, it evolves, it helps in evolving to overcome that particular stress. So, however, this is anybody will turn around and say this is an ex vivo infection experiment, it does not have much of a relevance. So, we performed this in an animal infection model. So, in an animal infection model, what we have done is we have taken the culture and we basically performed a aerosolic infection in guinea pigs and allowed the infections to prevail for a certain particular point of time. And eventually we took out the CFUs on day 1 and day 56 and looked at what happens. So here is the data. So day 1, the guinea pigs were infected with a similar level with either the wild type, the mutant complemented with the wild type, mutant complemented with the mutant. But what is important is 56 days post the infection, you can see that the mutant shows, this is a log scale, so approximately 3 to 5-fold higher survival compared to the wild type, because the stress here is the host-induced stress. When you put back the MUT-Y, then it is again back to wild type levels. When you put back the MUT-Y which has mutation, again showing some level of survival advantage. That was seen both in the lungs as well as in the spleen. So this more or less clearly underscores the point that whenever you have mutation in some of these DNA repair genes, then you would actually end up getting some level of growth survival advantage even in the host. To see how much survival advantage it is getting, we actually competed strains. We had put in a plasmid in RB, which is kanamycin resistance, and competed against RB MUT-Y, which is hygromycin resistance here. So kanamycin and hygromycin. So we can actually play it on either plain antibiotic plates or on kanamycin or on hygromycin. And then from there we can back calculate and see what is the % survival in each case. So we competed wild type with the knockout, competed wild type with the knockout complemented with the wild type, knockout complemented with the mutant. So this is how 3 competitions were done in guinea pig experiments. After that, we basically looked into the total number of CFUs we got on a plain plate. You can very clearly see that the total number of CFUs were very similar, irrespective of in lungs as well as in spleen. That is basically tells you about the total load of the Mycobacterium tuberculosis. Then we did the experiment wherein we looked into the computation. And day one, so in each set, this is the set here. So in each set, you can see that in day one, the load was very similar to RV competed with RV delta multiply or RV when RV is competed with the complemented. All of them are looking similar on day one. However, 56 days post-infection, you can already very clearly see one thing, that wild type does not survive as well as the mutant. When they are competed against each other, there is approximately 30-70 split. And when you put back the MUT-Y, that particular split goes away. And when you put back the MUT-Y with a mutation that is there in the DNA repair gene, we found in GWAS studies, we get this particular advantage all over again. And the similar thing was also observed in spleen, where we see this particular advantage when you have a DNA repair gene mutant. So this is basically what I would kind of summarize the study. We have other data too, but I will try to stick to one story and summarize the study. What we found out is that from the GWAS analysis, we have not identified any mutations in the DNA repair gene in the susceptible sequence, drug susceptible genomes. However, specifically in MDR, XDR, TP, which are 422 genomes, we found mutations in DNA repair genes and in both base excision repair pathway and NER pathway. We selected base excision repair pathway, NER pathway, and the mutations that were identified in GWAS were validated in Specmatis and found to be functionally compromise the activity. So now, what we believe is that no matter what the stress is given, it can be host-induced stress, it can be antibiotic stress or whatever, having mutations in some of these DNA repair genes helps in higher mutation rate, higher ex vivo survival, and higher in vivo survival. So based on these things, we basically think that DNA repair mutations in the DNA repair genes predisposes the strain towards evolution of higher levels of drug resistance in antibiotic, when antibiotic stress or host stress is given. So you may ask, if that is what is happening, why are the DNA repair genes are supposed to be important for our survival, right? No, some of the important ones which are absolutely necessary for our survival were never identified. These so-called DNA repair genes we have identified seems to be some of those things which tuberculosis can get away with. Deleting them or not compromising their activity. And whether it is widely used or not is something that we need to find out. But we personally believe that it is the first way. Our hypothesis is it is the first set of mutations which will eventually lead to additional set of mutations. We are testing this hypothesis by looking at the genomes of all those genomes where we found the DNA repair gene mutations to see if they have accumulated other mutations over a period of time. So, I have other stories, but I would stop because I really don't want to take any further time at this point. Thank you.
Thank you, Vinay. I do believe, because we just published a paper in malaria that DNA and most of the drugs when we use like artemisinin, DNA damage repair protein DD1 has been very important in that. So I personally feel.
I can't hear you.
Okay. Now you can.
I can't hear you. You're muted.
Okay.
Pawan, you're muted.
I'm doing it. Okay. I personally feel that this data is quite interesting because we just published a paper that the DNA damage repair protein DD1 in Plasmodium is actually linked to artemisinin mode of action.
Excellent.
So I do agree that many of these genes are associated with, because many of the drugs which we use against these pathogens, they cause DNA damage, and the parasite has to mutate this DNA repair protein to take care of that DNA damage. So I think this is really a story which is happening, and that's why many of these proteins are mutated so that to take care of the attack of these drugs. So I do agree with what you have presented very beautifully. And this is the first time I heard that DNA repair, role of DNA repair in the drug resistance. Is there any question from audience? So, any question from audience? But fantastic talk, and first time I heard your story. Thank you very much. It's beautiful. Thank you. Thank you for accepting the invitation.
There are something that is coming.
Yeah.
There is a question.
Okay, mutant, there is a question. The mutant generated upon antibiotic pressure, then by what mechanism, or are they enriched when the drug susceptible ones are cleared by chemotherapeutic intervention?
Are mutants generated upon antibiotic pressure? Okay. Or are they enriched when the drug susceptible ones are cleared by chemotherapeutic intervention? So we all know that majority of the time there is a problem when we don't consistently take antibiotics, right? We take antibiotics, but we stop antibiotics the moment people feel better. So there is a compliance issue. So that actually helps in evolution. Like, what happens is when you are consistently taking antibiotics, you are likely to eliminate all the bacteria over a period of time. In case of tuberculosis, unfortunately, it happens to be 6 months, right? But somewhere along the way, after 3 months, when people start feeling better, they kind of start becoming more sporadic intake of the antibiotics. And that essentially means you're leaving a residual population which can evolve. And mutations are spontaneous. The pressure is there, but pressure is not consistently there. So the antibiotic concentration goes down. And you are ready to, you generate 1 or 2 of those mutations over in that particular time, correct? That helps in evolution. So these are spontaneous mutations, and compliance issue is one of the problems.
I fully agree with this, because this is the way things, I personally feel that many of these drugs are antibiotics. They are creating the DNA damage for the pathogens or this one. And so they have to evolve to take care of that. I think your approach is the real right one. Thank you. Thank you, Vinay. It has been very nice. Thanks a lot.
Stewart, can you hear us?
I can hear you, yes.
Great. Nice to see you. So, participants, it's time to shift gears and move towards some demo and practicals, as we have been doing last 2 days. So, to start with, we have a lecture by Stewart Clark, who is a senior application scientist in Cresset, Cambridge, United Kingdom. So Stewart obtained his PhD in chemistry from University of Bath, where he investigated the use of computational methodologies to study chemical reactivity. He then spent 20 years working for small biotech contract research organizations, where he was responsible for developing and implementing their computational chemistry services to prospective and existing customers. Stewart is now a senior application scientist at Cresset. His responsibilities include training and supporting Cresset customers so that they can achieve their scientific goals using Cresset ligand-based, structure-based software solutions. So today we will learn about a very important aspect of how to deal with structures of protein, how to design drugs, which is a very important area in drug designing against infectious diseases. So over to you, Stewart. Thanks for joining us.
And thank you for that lovely introduction, and I also just like to take a moment to thank the organisers for allowing us to present here today. So thank you so much for that. Before I start, I think I should just mention that ideally what we would have liked to have done is provide a case study towards something that is towards infectious diseases, TB and malaria. Due to time restrictions, we were unable to get something ready for that. So I hope you will enjoy today that a sort of a more general introduction to the Chrysalis software, which I'm going to be giving you. And so what I'm hoping to do today is going to be sort of like two things I'm going to do. I'm going to first of all give you a 30-minute introduction to the Chrysalis software, and then for the following 90 minutes, I will be giving you an introduction to one of our soft pieces of software, and showing you some basic concepts of using it to use it in a drug design environment. So what I'm going to do now, I'm going to now share my screen, and hopefully you'll be able to see that in just a second. Okay, so hopefully you can now see my my slide deck. If you can't, then please shout out. But I'm going to assume that you can. So what I'm going to do then is first of all give you an overall view of the Chrysalis software solutions. I'm then going to give you a brief introduction to the Chrysalis science. So, underpinning all the solutions, software solutions that we have a sort of fundamental science which is used to generate some of the CAD solutions which we offer. So, one of the key pieces of software which we're going to be looking at in a little bit more detail is Flare, and that gives you the ability to look at things from a structure-based perspective, that is, where you have a protein structure that you can design into, or a ligand-based perspective, which is basically when you don't have any protein structures, but you may have some known active ligands, and you want to sort of hypothesize a model based on those known active ligands to try and find some new active ligands. And then following that, I'm going to very briefly introduce some other aspects of our software, Spark, Blaze, and Picker. All of these have slightly different viewpoints of what you want to do in the drug discovery arena. So I'll briefly touch on those as well. And then after that, I will move into the Flare demo. So before I move into the Chrysalis science, let me just give you a brief view of the company. So Chrysalis was founded in 2002, and we basically have two arms towards to the company. The first arm is the software side. So we design, program, and sell software for drug discovery purposes. And we have software within 15 of the 20 top pharma companies in the world. And in addition to that, we have a lot of other customers across the world, including North America, India, the EU, China, Japan, and Korea. And so that's one side of the company. The other side of the company is a sort of contract services side. Chrysalis Discovery has, in its lifetime, worked on over 300 projects, delivered to various customers globally, and so that's still a very active area for us as well. And so through both the services side, Chrysalis Discovery, and our software side, we're able to support medicinal and computational chemistry through ligand-based and structure-based design software and services. We're a growing company. So year on year, we've got some rise in 20%, which is really great growth. So we now sort of have 50 plus staff across 3 different continents. So here is kind of a brief overview of what we offer as in terms of the software. So the main platform that we have for ligand-based and structure-based design is FLAIR, and this is what we're going to be looking at in a little bit more detail later on. And so that encompasses both ligand-based and structure-based design. We also offer a platform for virtual screening. So virtual screening is when you basically have a starting point, maybe a ligand or a protein, and you then have a database, a large database of molecules which can be potentially purchased. So those molecules can be virtually screened, so they're sort of in the computer, if you like, and by using the existing information that you have, either based on the protein structure or known ligand structures, you can then sort of screen millions of compounds, and we offer that either as a service, and it's rather than being a desktop program, it's actually a server-based program. So because you're dealing with lots and lots of molecules, that has its dedicated server. In addition to Blaze, we have a program called Spark, and what Spark allows us to do is it allows us to look at an existing molecule and perhaps gives us a chance to change that molecule in some way. Maybe the molecule is not we need to sort of develop a novel molecule, or perhaps that molecule has some issues with pharmacokinetics, so we need to change some of the functional groups within that molecule. Then Spark allows us to do like an automated replacement of parts of that molecule to come up with some new ideas. And then finally, at the bottom of the slide here to the left, we have a program called Picker. And what Picker allows us to do is, if you're designing a library of molecules, so that say you're dealing with a specific sort of chemistry reaction, and you can then buy a number of reagents that sort of go through that chemistry reaction. What we can do is we can design. You can say, well, actually, I only have the ability to make 200 compounds. So what reagents am I going to select? You'd probably want to select ones which are the most diverse in terms of their features. So what Picker does is it basically helps you to select a diverse set of molecules. So now I've just given you a big introduction. Let me sort of start to talk about the Cresset the science behind the software at Cresset. And so there's all impinges on something called a force field. Now, force fields in general in computational chemistry are basically ways where you can describe molecules using basically a basic description of how those atoms relate to one another. And atoms, so you obviously have different types of atoms, so you have different elements like carbon, nitrogen, oxygen, and so forth. But in addition to that, those atoms find themselves in different environments. So you have to have different types of carbon atoms, different types of nitrogen atoms, different types of oxygen atoms, depending on their local environment. So actually generating what they call a force field is actually quite a complex thing, because you have to have lots and lots of different atoms types to effectively describe your system. But the advantage of this is that they are quite fast, which means that you can model simulations using protein, involving proteins, and also small ligands as well. But what our founder found, Dr. Andy Vincer, was that some more discrete interactions that involve the interactions between two different ligands, for example, are not well described by these general force fields, because what happens is that each of the atoms is assigned a partial charge, if you like, and so these partial charges are good for general purpose modelling, but when you want to model sort of very specific interactions, they they are too approximate. So what I found to do was basically said, well, actually, what we're going to do is separate the pi, like the pi charges from the atoms from the sigma charges, and we basically put the sigma charges onto the atom centers, and then we add, if you like, virtual p orbitals, what we call z atoms, to describe the pi charges. And doing this approach allows us to begin to look at sort of how substituents can start to affect the electrostatics of a molecule. So, for example, in this particular case I've shown here, we've got sort of two chloro groups being attached to an aromatic ring, and they tend to pull charge away from the aromatic system. So what you can see here is that the negative electrostatics are being pulled towards the chloro group, so that you've got this blue negative region, and then where the negative electrostatics have been pulled away, you've then got the positive region sort of on the far side there. And then the advantage of separating these charges is that you can start to better describe how charge is surrounding your particular atoms, what we call charge anisotropy. And this allows us also then to start looking at more subtle interactions, like what often we call pi stacking interactions. So where you've got two aromatic rings that are close to one another, they can interact with one another based on how their local electrostatics are with respect to their environment. So what Z force field represents is that it's a high-quality molecular mechanics force field that can be used for small molecules, waters, and proteins. So here is a sort of an example of what we kind of generate with the force field. So what you're seeing here is a 3D molecular electrostatic interaction potential of a ligand molecule. And so this is essentially, if you like, a protein's eye view of what it sees when the ligand binds. And so you have like a sort of the surface over the ligand, where the red is the positive regions, the blue is the more negative regions, and so you have this very rich, information-rich surface which the description generates. But when we look at modelling, what we often want to do is look at lots of different molecules, lots of different ligands at the same time. And what we find is that by if we try to compare those ligands with the 3D field that you see here, we find that it's too computationally intensive. So we have to think about doing this in a slightly different way. So what we do is we basically sort of distill down to the most salient features of the surface. So we kind of identify the maximum the points around that surface that are maximum in positive electrostatics, the ones that are the maximum points of negative electrostatics, and we also identify key areas for sterics and for hydrophobics as well. And this generates a sort of 3D set of data points called field points, and it is this that we use to when we look at our sort of ligand series that we're going to look at in the program. So at the moment we were talking about ligands, but we can also extrapolate the charge description to proteins as well. So not only can we describe the electrostatics of a ligand, but we can also describe the electrostatics of a protein as well. So again, on the left-hand side here, we have the protein with the surface where blue is negative electrostatics, red is positive electrostatics. And its corresponding ligand is on the right-hand side here, again showing where the negative and positive electrostatics are for that particular ligand. So another sort of viewpoint of that is rather than sort of having to compare the electrostatic of a ligand to the electrostatic protein at the same time, what you probably want to do is to maximize the binding. You want to try and make sure that the electrostatics of the ligands are complementary to the electrostatics of the protein. So we've got sort of more negative electrostatics in the ligand, you want more positive electrostatics in the protein, and vice versa. So one thing that we do is, as well as looking at colouring it in terms of what electrostatics we have, we can colour it in terms of how complementary are the electrostatics in the ligand compared to the protein, and vice versa. And we use a colour scheme for that, where green represents good electrostatic complementarity, and red represents an electrostatic clash. So let me sort of give you a brief overview of FLAIR. So FLAIR represents a platform that allows you to do both ligand-based and structure-based design work. We have a number of advanced tools. We've just briefly talked about electrostatic complementarity, and I will deal with that a little bit more in the demo. But we have more advanced tools as well, like free energy preservation and force fields and molecular dynamics. These are quite quite intensive things, but are available to people who want to do that. And the focus of FLAIR is a very ligand-based, ligand-focused program. So it allows us to look at things like ligand-based alignment, docking and storing of ligands in proteins. You can set up pharmacophores, so that's when you've got no protein information, but you've got some ligand information, and you can use that to generate a hypothesized immune model. And then because FLAIR is based on Python as a language, it means that it's easily added to by users if they have Python experience. And in addition to that, we also offer nodes for Lime and Fragment Pilot as well. So let me move on to just some general features of FLAIR. So one of the things that you have the ability in FLAIR is to, if you've got a protein structure available and that electron density map is available as well, you can download that electron density map so that you can then view that in the program to just double check that, for example, your ligand exists in the protein structure, and that allows you to just double check that you're modelling the correct thing before you start doing quite computationally intensive jobs. The other thing you can do is you can also edit the protein. So very often in the PDB you get lots of different alternative conformations of side chains of residues. And so what you can use the program to do is basically analyze the alternate conformations that are defined in the PDB and then select those, and then go on to prepare the protein ready for further simulation. One of the key aspects to molecular modeling is being able to dock ligand into a protein structure, and we have a module that can do that. And when you dock a ligand within a protein, what you get back is, if you like, how well that ligand fits that particular protein active site by a score, and basically the better the score, the more interactions that ligand is making with the protein active site. And in addition to the more sort of traditional docking approach, you can also do covalent docking. So very often protein structures bind their ligand through a covalent interaction. either through, it's usually through a cysteine or serine or threonine side chain in the binding site. And so you can use FLAIR to look at sort of covalent docking in those particular situations. The other thing you can do is consider the flexibility of the protein by defining different sort of protein snapshots in FLAIR, if you will, and then when you start the ensemble docking, the part of the docking routine will basically select which of these docking snapshots are used to dock the ligands in. And this is useful if, for example, you have, for example, a loop in the ligand which is in a slightly different place in one crystal structure compared to the other, and you're not quite sure which one would be appropriate for the ligand. In fact, you may have ligands that bind to one situation or the other situation, so you may need to sort of incorporate both those snapshots and allow the program to decide how best to dock the ligands based on which snapshots it uses. So the other thing, so docking is one aspect to what people would do with molecular modelling. The other thing that you'd perhaps want to do is align your molecules if they are very similar to one another. So you may have, for example, a ligand in the crystal structure, and you then have a number of other ligands, but maybe they have 2D, they have a 2D representation. So what you can do in FLAIR is convert those to 3D, generate the conformations, and then align those conformations with what we call the reference molecule, so that they all align in the same coordinate frame. And that is useful for when you want to go on and do sort of more advanced calculations within FLAIR. And those alignments can be done through perhaps one of two ways. One, you could do if the structures are very similar to one another, you can do that through a substructure alignment, or if your molecules are different. So if you know that you have different ligands that bind to the same binding site, and but they are completely different in their structure, then another thing that you can do is align them using, rather than substructure, the electrostatics and shape compared to the reference molecule. So what this allows us to do then is, what the user can use that for then, is to basically help them design new molecules which look different, and also be able to virtually screen a few thousand molecules on their laptop. So one of the things that I sort of briefly touched on the introduction is the ability to use FLAIR to look at the electrostatic interactions between the protein and the ligand. So here's another example of streptavidin and biotin. And again, what we can do is basically colour things either by the electrostatics, which we have on the top here, or by the complementarity of the ligand and the protein electrostatics. And what this can do is it can allow you to say, well, actually, in this region here, we have an electrostatic clash, it's coloured in red. How can I edit my molecule so that this red colour turns more green? So you can use it in a way which allows you to give you a bit more insight of how to perhaps design a new molecule. And the other thing that's it, so we've been focusing quite a lot on structure-based approaches. We can also look at ligand-based approaches, and what you can do here is so what we can, in fact, but in this particular slide, what we're doing here actually is we're actually comparing. So we've got two quite similar molecules here. One on the left-hand side here, which is less active and more active, and one molecule on the right-hand side, which is less active. And what you find is that you can start to look at how the electrostatics are different for the molecules on the left-hand side compared to the molecule on the right-hand side, and you can start to try and understand where those differences are, and perhaps how you edit those, perhaps the molecule that is less active to make it more active. So in this particular case, we have on the left, so we have the molecule which is more active has an oxygen, so it's on that 5-membered ring has oxygen on that 5-membered ring, which is an oxazole. And you can see here that next to that oxazole, you have this sort of green region in this electrolytic complementarity surface. So that shows that it's complementary with the protein. Whereas on the molecule below, which is slightly less active, you can see that actually instead of the oxygen in the ring, it has a carbon, and so that carbon is going to be slightly positive, and so obviously in that region of the protein is also slightly positive as well. So you've got more of an electrostatic clash there, so it turns red. So what this allows you to see is where you may have room for improvement for your particular ligand. Now let me turn to ligand-based methods. So ligand-based methods occur when, for example, there's always quite a lot of projects out there which don't have protein structures, and so when that happens, you can then say, well, what do I have that can help me inform me of perhaps a model that I could use? And one of the things that you can do is, if you've got lots of ligand structures with known activity, you can use those to build a hypothesis based on the activity of those ligands, and you make the assumption that those ligands are binding in the same place. So you do an alignment of those ligands, and that allows you to provide you with a summary of the landscape of how those ligands align onto one another. And in Flare, what you can do is it provides you with sort of regions in space which perhaps, if you put electrostatic in a certain region, it basically hinders the activity. If it can show you sort of regions in space where adding hydrophobics might be detrimental to activity. And so it does this by using the activity data of the known ligands, and then sort of cleverly sort of saying, well, actually, where these ligands are less active, this is what's happening. And so if I keep, this is a region where if I have the positive electrostatic, I will gain activity. If I have the negative electrostatics here, then that will also be an improvement of activity. So this is called Activity Atlas, and it allows us to give us a brief overview as to what sort of features would be useful in a molecule. And then on the right-hand side here, what I've also shown is that as well as doing a hypothesis type of calculation, it also allows you to look at the data that you have. So you may have a number of molecules and their associated activities. And so what you can do is you can compare the activity between pairs of molecules to see what sort of groups are increasing the activity, what sort of groups are taking the activity away. And also, more importantly, is that you can start to look at sort of chemical similarity, if you will, and how similar molecules may have quite significant changes in the activity. And this is often called an activity cliff. And so what this allows you to do is sort of focus in on small changes which perhaps lead to very large changes in the activity. And so what FLAIR allows you to do is it allows you to sort of analyze that data and sort of focus in on these sort of small changes that may represent an improvement or detrimental, or it's detrimental to the activity. So in FLAIR, what you have, ligand-based approaches, is the ability to bring in a number of different molecules and show things in a slightly different way, which allow you to look at how similar the molecules are and how their activity changes as a result of that. You can look at pairs of molecules and identify the points where you've got the most steep activity cliffs. And you've got a number of other views which allow you to view how these molecules relate to one another in terms of their activity. So another thing which you can do from a ligand-based point of view is what they call QSAR methods. And so what this allows us to do is again to identify sort of, the features in the molecule which are positive regions, negative regions, highly rapid, and so forth, and you can use it to build a model to predict your activity of your ligands. So that, again, this would need a number of known ligands, and then using the alignment tools in FLAIR, the program will then subsequently go and generate a QSAR model, which allows you to predict the activity of molecules that are yet to be made. So another thing that you can do with FLAIR is to generate pharmacophores. So if you have a number of ligands which have a good activity and you think they are bind to the same part of the binding site of your target, then what you can do is you can bring those ligands into FLAIR, and basically what FLAIR does is it basically aligns them and comes up with a hypothesis of what we call a binding site hypothesis of how those molecules might look if they are bound to the target. And you do that if you would do this if you did not know the information of your protein. So basically, so you have lots of different options in FLAIR, and so the sort of take-home message here is that it allows users to look at both ligand and structure-based methods, which you can work in synergy of that. So if you've got some extra crystal structures for proteins and you've got some ligand information, you can use both ligand-based and structure-based methods to help you guide you through the molecule design. We've tried to make the interface as easy to use as possible, so hopefully that will help people sort of gain confidence in using the software. And you have the ability to do quite a lot of local processing, so you can display up to 100 proteins simultaneously and in stereo. Before I go on to the demo, what I'm going to do now is just briefly introduce you to our other software offerings, Spark, Blaze, and Picker. And just starting off with Spark, so as I mentioned briefly right in the introduction, Spark is set up so that if you start with a molecule which you don't really like, maybe because of IP issues or maybe because of pharmacokinetic issues, Spark is there to help sort of come up with some ideas on how to replace parts of that molecule with it suggests a new fragment to add to that molecule to come up with some new molecular ideas. So this will be something like scaffold hopping, for example, or you may want to say, well, actually, I want to generate a small library of quite similar molecules, R groups. I want to explore the SAR around that. And so you can do sort of an R group exploration in addition to scaffold hopping. And so what you can do in terms of the more complex experiments is you could actually bring in 2 molecules and say, actually, I'm going to edit molecule 1, but I want it to more like, I want the solution to look more like molecule 2. Or if you've got 2 fragments in different parts of the binding site and you want to link them together, then you can do that as well. And there are a number of options, like, for example, trying to place water molecules within the binding site so that your molecules makes those interactions that the water molecule was once making with the binding site. So so again, it uses the Z force field that Cresset has, and what it does is basically when the new fragment is added, so the fragment on the bottom here is the bicycle instead of the cycloketone at the top. So what the program is doing is, rather than comparing the fragment similarity between the 2 molecules, it's comparing the electrostatics and the shape of the whole molecule. And the advantage of that is that the new fragment actually has an effect on the rest of the molecule electrostatically in terms of the way in which it pulls electrons from the rest of the molecule or donates electrons to the rest of the molecule. It has an impact on how that molecule looks to the protein. So what we need to do is think about it from a molecular, a whole molecule perspective rather than just the fragment perspective. And so what the program does is to consider things as a whole molecule and score things as a whole molecule. And that means we can take into account what people call neighboring group effects. So it ensures that the electrostatics of what you're generating is very similar to the electrostatics of what we're wanting to replace. So the other input into Spark is fragment databases. So it needs a fragment database so that it can sort of replace the bit of the molecule that you want replacing. And what the program has is an input database of commercially available so fragments coming from commercially available compounds. So institutions like e-molecules, for example, have lists of molecules that can be purchased from various suppliers, and these have been fragmented down to around 7 million fragments, and these 7 million fragments are then you can then use those to come up with some new idea for your molecules. The other thing that you can do, the other sort of fragment type we have is commercial reagents. So very often you may have a specific chemistry in mind, and that you want to just restrict yourself to that specific chemistry. Maybe it's adding an acid to an amine, for example, or sort of adding, using boronic acids. So we have a number of commercial reagents available where you can select that particular database and come up with some solutions which only just consider that particular chemistry. And finally, if you have any sort of proprietary information yourself and you want to keep it in-house, then another option that you can do is you can create your own database so that that will sort of decompose your database into fragments and then use that as part of the process. So the advantage of Spark is that it basically allows you to generate new ideas for maybe something where you need new IP, and it can give you obvious results, it can give you less obvious results, but it can also give you very novel results as well. And so hopefully the solutions that come out of Spark allow you to get some new IP, give yourself a new IP position, give you some new ideas on how to move forward with the project. And the other thing which the program does is it allows you to share ideas with colleagues fairly easily. So we have a number of things that you can use to help describe what you're looking at with your colleagues, and we also made it simple to use so that there are sort of 5 options when you open the program in a wizard kind of way, so that you can say, oh, I want to do a scaffold replacement, or I want to do a water replacement. You can just click on those examples, and then the program helps you set that up. So you don't have to worry too much about how to set the job up. So I want to now move on to Blaze briefly, and Blaze is basically the virtual screening option that we have in terms of it's rather than being a desktop program, it's a server program, because again, what you're dealing with is wanting to briefly look at want to identify molecules which are very similar to one that you already have. So you would use this maybe if you may only have 1 or 2 molecules and you want to get some more hit matter, for example. So maybe, for example, we've got this steroid just here. What we can do is what we can do then is define that steroid in terms of its electrostatic, its hydrophobics, in said force field, and then what we do then is we then take the 3 million commercially available compounds, and we say which of these 3 million commercially available compounds look similar to, which have similar electrostatics and shape information compared to the steroid that we already have, and it will then go on to basically score those and show you the ones which look the most similar, and you could then perhaps select a subset of these that are chemically diverse. So you'd go to this chemical supplier and purchase those and test those in some assays to see if they have any activity against your target. Finally, we have Picker. So Picker basically gives us the chance to, so we have, the restriction here would be, I only have the ability to test 1,000 compounds, but I want to explore the SAR of that series, and but I want to do it so that I rather than look at things that are very similar, I want to explore the areas of chemical space, so that I want to ensure that the monomers that I select cover a large area of that chemical space in terms of their 3D representation. So very often what people do is they will look at this from a 2D representation and select the monomers that look the most different. But that is only taking a 2D representation of that into account. If you wanted to, what Picker does is it basically looks at the electrostatics and the shape of the monomer and tells you from that perspective what is the most different. And so you have the ability to, when using Picker, to basically come up with a series of monomers that not only are different in terms of their 2D representation, but also are different in terms of their 3D representation based on their electrostatic and their shape. And this is probably sort of concludes the presentation part of what I'm going to do, what I'm going to show you today. And what I'm going to do now is I'm going to now move into Flare, and what we're going to do for the next 60, 90 minutes or so is I'm just going to show you some introductory features of the program so that you can understand how you can perhaps set something up. So let me escape my slide deck here. Minimize that. So hopefully you can now see my copy of Flare, which I've already got open on my computer here. Again, if I'm hoping that is the case. What I'm going to do is, excuse me. What I'm going to do is I'm going to now open a going to download a PDB file, and what I'm going to do is download a PDB directly from the RCSB. And what I'm going to do is I'm going to do I'm going to download a PDB file for RIP kinase, and so it's a kinase structure. And what I'm going to do is, to do that, I'm going to put in the accession code for that. So it's 5HX6. And the other thing that, as I mentioned in the presentation, you can download the electron density maps as well. So if they exist, so what I'm going to do, they do exist for 5HX6. So I'm going to download the electron density maps, so I can show you that in a moment. And then I'm going to push OK. So the next thing that the program asks you is what kind of preparation you want to do in terms of the protonation state. So you have available to you 2 or 3 options. So you have use input protonation state, which basically means that it will leave things alone. So if you've got a situation where a colleague has prepared a protein for you, that is probably what you would want to do. But as we are going directly from the PDB, we're going to do a full protein preparation, like so. And push open here. So that brings down downloads the protein structure, and the one final thing before we prepare the protein is the protein starts to add. Before we prepare the protein, the program begins to needs to ask a couple more questions. One is to so cap change is basically so in some proteins what you find is that in some crystal structures is that the very flexible regions of the proteins, they can't resolve the protein structure well. So what they need to do, so what you find is that there's parts of the protein structure which are missing. And so what a cap change does is it basically asks you what you want to do with the backbone, where the backbone finishes, and in that missing section. So what it does is it basically, instead of leaving it bare, it basically caps the chain in terms of adding an acetyl group or an N-methyl group. You then have the ability to extract the ligand. So if there's a ligand available in your crystal structure, what's going to happen is you're going to extract that ligand and put it into the ligand table, which is just on my left-hand side here. And you also define what you want to define as the active site. So by default, this is 6 angstroms, but you can change that if you think if you want it to be a bit more or a bit less than that. And then you can remove, so obviously many proteins have water molecules as well, so you can decide whether you want to keep all the water molecules in the protein or just keep the one within the binding site of the protein, of the active site of the protein. And then finally, you have the ability to copy proteins. So what this does is it leaves the original protein intact, so that when you finish preparation and you may need to go back to the original protein to prepare it in a slightly different way, at least you can then do that if you don't overwrite it. So copy proteins is is selected by default. So we now push start to prepare the protein. So that only takes a few seconds, but as that works, let me just sort of guide you through what we are seeing at the moment. So on my left-hand side here, we have a ligand table So that anything to do with ligands goes into this table here. So if you bring in some ligands using an SD file, for example, they will all go into the ligand table here. Just at the bottom here, we have the proteins window, and the proteins window has everything that isn't a ligand essentially. And you'll notice here now we've got the original 5HX6 protein and the prepared protein, which is given as a suffix _P. And then we have a number of other windows, which we will talk about in a little bit. But we'll also have the main 3D view window, which is in the center here. And by default, what you see here is the protein structure is given as a ribbon, so sort of secondary structure cartoon, if you like, so that the alpha helices are in sort of an orange color, the beta sheets are in a yellow color, and the loops are in a green color. And the atoms in the binding site are shown, we'll see those in just a second, but all the other atoms are hidden away, although they do exist. Let me just show you is that Flare also has a number of tabs. What we've tried to do is make it relatively easy to find things. So if you wanted to do something with a ligand, you go to the ligand tab. If you want to do something with the protein, you go to the protein tab. The home tab basically gives you everything which you would use fairly regularly, like visualization or changing the visualization of the molecules. So let me now sort of click on one of the ligands here. I should just mention that 5HX6 is basically a dimer. So you have an A chain and a B chain. So you have two ligands, and ligand associated with the A chain and a ligand associated with the B chain. And what I can do now is if I click on the A chain ligand and push the focus button, I should focus in on the ligand like so. So what that kind of sort of zooms in on the region that I want to have a look at. And what I can use is I can then use my mouse button, left-hand mouse button, to rotate my ligand, so my system, and I can then use my middle mouse wheel to zoom in and out like so. And the other thing which you can do is basically use the features in the home tab to control what you see. So at the moment, we're seeing the protein in the ribbon form. If I click on the pull down here, you can see that the cartoon is selected, but you can also look at it in the sort of C-alpha trace tube or plat, or you can turn the ribbon off altogether by just clicking on the button just here. So in terms of the ligand, we can change how we view the ligand. So if I double click on the ligand to pick it, then you have a blue halo around that ligand. And if you go to display, you have a number of different ways, and you'll find that with many modeling programs you have this. You can change the view of what you see. So at the moment we have something called cap sticks, which is like a thicker stick. But you can also do lines, which is actually what the protein is currently shown in. You also have thin sticks. You can also have baller stick, and you can also have CPK, which is like an approximation of the true volume of the atoms within the molecule. So let me go back to capstick, and what I can do now is if I go to the other thing that you might want to do with protein ligands is to look at the interactions between both the ligand and the protein. And so in the home tab just here, you have a contacts button, and that contacts button basically shows you the contacts that are taking place between the ligand and the protein. And those dotted lines represent different things. So different interactions have different coloured lines. The green line you've seen there, that's a hydrogen bonding interaction between this carbon oxygen here and this nitrogen here in this residue here. And the purple line represents can represent a number of things, but in this case it's a pi stacking interaction between the oxyl ring in the ligand and the phenylalanine side group here in the shown just here. And the reason why this is looking like it's going into space is because I think it's hitting the hydrogen in the phenylalanine. So what you can do is if you push the H button, oops, let me just unselect the ligand. Let's try again. So let's if I double click the phenylalanine and push H, you can then see that when I've added H, so the hydrogens which are non-polar are hidden away by default. If you want to see all hydrogens, then simply push the H button here. And so what I can do here is I may think, well, actually, I want to highlight these residues, which means making these interactions with in a little bit more detail. So what I can do is I can double click on. I should just mention that before I double click on it. When you hover over the atoms, they bring up this pink halo. Now, if you hover over with the pink halo for a couple of seconds, you get a tooltip, and the tooltip says what that atom belongs to in terms of the residue. So you know that this is phenylalanine 162, and that is an aromatic carbon. And what I can, so if I click once, I click a specific, I pick a specific atom. If I click twice, I pick a whole residue, so that's the whole phenylalanine residue. And at the same time, I'm quite interested in this other residue here. So if I hover over this nitrogen, it tells me that this is ASP156, so this is the backbone nitrogen for that. And I want to select it in addition to the phenylalanine. So what I'm going to do is I'm going to push Control on my keyboard and double click on the aspartate, and that adds to my selection. And what I can do then is with those two residues picked, I can then transform these into perhaps something slightly different, like a thin stick. And so this highlights those residues for me that seem for me a bit more important. And so by default, what you're seeing is the interactions between the ligand and the protein, and ligand and water molecules, if the water molecules are there present and interacting with the ligand. In this case, that's not happening. But you can decide what you want to see further. So if you click on this little contacts option, what you can do is you can start to define what contacts you want to see. So at the moment, as I say, we just see contacts that are involving the ligand, but you may want to see contacts that are involving interactions between protein-protein atoms, for example, or interaction between protein and water molecules. So we have this sort of matrix of checkboxes, if you like, and so what we need to do to highlight these interactions is just simply check the checkbox. So in this case, this highlights the interactions between protein atoms and protein atoms, which you can see in what's happening in the background there. And if you want to highlight, if you want to put a little bit more emphasis on the protein, maybe the protein-ligand interactions, what we can do is we can actually click on this checkbox again to give a thicker square, and this highlights the dotted line into a thicker line just here. And so you are able to sort of emphasize features, molecular features, which seem quite important. And also, if you want to turn off, then you simply just toggle the checkboxes, so they sort of toggle between off, on, or thicker lines. So I'm going to now do the same with the protein. I'm going to turn off the protein-protein atom interactions. So now we're just seeing the ligand interactions again. Push close. So what I'm going to show you now is I'm going to show you how you can edit a molecule within Flare. And what I'm going to do is I'm going to select the A chain ligand here, and I'm going to go to the ligand tab and go to edit, and then I can edit a copy of the selected ligand. And then what happens then is that a new tab is generated called editing tools. And what this is kind of a sort of a sketch pad, a 3D sketch pad, if you like, which allows you to change some aspects of the molecule which you're editing. So when you go into that editing tool, by default, everything else which you see, so the protein, for example, sort of goes into it's grayed out. So whenever you're editing the ligand, the protein is not going to be affected. It's only going to be the ligand which you've asked to be edited. And so what I'm going to do here is I'm going to change this isoxazole, and what I'm going to do is I'm going to change it into a slightly different ring. So what I'm going to do, I'm going to delete the oxygen and nitrogen. So if I hover over the oxygen, I'm going to push delete on my keyboard. That should delete the oxygen, and then I'm going to do the same for the nitrogen as well. What I'm now going to do, with carbon selected in the elements and fragments, I'm going to now use my left mouse button and hover over this carbon here, click on my left mouse button and pull down to generate a new bond to a new carbon atom, and I'm going to repeat that another couple of times. I'm then going to connect the ring together. I think that's right. So as you can see, that's not very well drawn ring. So we'll deal with that in just a second. The other thing which you can do is if you think you've made a mistake, you can do an undo and redo using the buttons at the top here. So this button here does undo. So if you feel that you've done a really poor job, you can try again. In fact, let's do that. So, and then because I'm going to convert this to an aromatic ring, what I would need to do is convert some of these single bonds into double bonds, and you can do this a number of ways. But one way you can do is if you hover over the bond so it turns pink, I can push 2 on the keyboard, and I will do it for this bond here as well. And then what you can do is, as you can see, it's still not designed, it's still not drawn particularly well. So what we're going to do is, if I'm going to use this lasso option, and I'm going to select the parts of the molecule that I'm just going to turn it around just a little bit more. I'm going to highlight parts of the molecule that I want to clean up because they're not very well drawn. So what I can do is push the lasso button, highlight the bit of the molecule that I want to clean up, push clean up. So, and what that does is it basically gives the parts of the molecule that I'm wanting to clean up an appropriate better bond lengths and angles, so that it looks a little bit better. Now, there's another option here which I'm not going to play with, but there's another option here called minimize. What minimize does is it also takes into account the environment of the system, so that if you
how can we remove hydrogen bond distance over bond line green to black?
That's a good question. So you would want the hydrogen bonds to black. So at the moment, that's not feasible. So at the moment there's a number of things that you can do in terms of changing the colours, but actually the hydrogen bonds is something that is written in the program itself. So at the moment we can't do that.
Can we remove hydrogen bond distance? That is 2.1.
So, I'm sorry, you're quite faint. Can you just repeat your question, please? Could you just repeat?
Sir, actually, the hydrogen bond distance, suppose it is 2.1 angstrom. I don't want to show this hydrogen bond distance. So.
All right. So you don't want to show the distance. You just want the dotted line and not the distance.
Yes.
Okay. So can we get rid of that? So at the moment, you can't do that in the current version of Flare. So you still need you can't actually get rid of the distances.
Sir, can we, one more question, can we do covalent docking complex for MD simulation?
I'm really sorry, you're really faint. Are you able to get close to the microphone somehow? So you're really faint.
Hello, Stuart. It's Sadaf
Hello.
here. Yeah. The question is basically, can we perform the molecular dynamics of the covalent docked complex? Is that the feature available in Flare right now?
So can I just double confirm what you just asked me? So can we do MD or covalently docked complex? Was that the question?
Okay.
Yes.
So yeah. So what you would do then is, that's a really good question actually. Let me think about it. I was going to say yes. What you would need to do is define the ligand as part of the protein. So this would be something that we would be able to help you out with if you were wanting to do that. It's a little bit more advanced, and I'm not going to show you that today, but that is definitely something that which we could set up. We could help you set up.
So, sir, we are able to remove hydrogen bond distance numbers.
Yeah, I was just trying to think about that, whether we could do that. I think at the moment you can't. You can. We can make. Let me just double check that, because we need to file preferences. Let me just see if we can. Okay. No, not general. Okay. Let's. So at the moment, it looks like you can't. So the hydrogen bond distances are kind of set with the dotted line. So you can't actually change the view. What you can do is, if I just push down on here for a second. I went through it quite quickly, but what you can do is so let me just see if we can. So show contest. So that's if you want to show contest, and then you basically have the ability to change which contacts you actually see. So at the moment, everything is turned on, but actually, you may only want to look at hydrogen bonds, for example. So you turn off all of these, but this doesn't do what you just asked me, which is basically you want to remove the label. We are working on something which I think will do that for the next version of Flare, but at the moment this is not possible.
Okay, thank you. Please continue, sir.
Thank you very much for your questions. I appreciate that. I'm now just thinking about the covalent question again. It might be problematic. I'm just thinking about it aloud, and it may be. So you can definitely do covalent docking, but doing covalent dynamics, one of the problems that you have is whether you can when you attach your ligand to your protein, is whether there are parameters available that define that interaction, and there may not be. So if it was a peptide, it might be okay, but if it's a novel ligand, you might have some problems, because for the molecular dynamics we use amber, and it would depend on whether there are the parameters available that will be able to define the covalent interaction. So I'm sorry, I was just thinking about it again, that it could be actually quite complex. But again, this is something which, if you're interested in doing, we'll have a look at and see what we can do. So what people will do is sort of email us at support. And if it's not me, then it'll be someone else who'll be able to help you out in terms of getting the simulation up and running if it is indeed possible. Let me go back to showing you what we can do in terms of editing the molecule. I just come out of the editing tool, which is good because I had just finished the molecule preparation, and so what you do now is once you've generated that, you can now rename this. So we can call this, by double clicking on the title, we can call this design 1. And then maybe I want to do another design which is very similar to the one I've generated. And simply another way of editing a copy is you can do right mouse click, edit a copy. And then what I can do then is maybe I want to make this carbon and nitrogen. So what I can do with this is hover over this carbon here and push N on my keyboard. And then push done, and then I can double click on the title and call this design 2. So one thing that's quite nice is the ability to divide your ligand set into different groupings, what we call roles. So we have one role in the ligand table called ligands, and currently has 4 ligands in that. So one thing that I might want to do is to move the designs to their dedicated section, dedicated role. So what I would do is I can select design 1, and I can then control select design 2, and then what I can do then is if I right mouse click and go set role to new role, and I create a new role called my designs. And what that does is it moves the design molecules to my role, my designs, and so you have as a user the ability to organize your ligands so that you can easily find them, and also you can drag and drop them into different roles. So if I want design one back into ligands, I can just simply take design one and drag it back into the ligands, or then drag it back into my design. So I have control over where I want my ligands to be in my ligand table. So the next thing I'm going to do is show you some how to set up a docking. Before I do, I just want to edit the protein a little bit. So the protein is quite a large system, and so I want to just make it a little bit more simple at the moment. So what I'm going to do is what I would like to do is to delete the B chain and just consider the A chain. And so what I'm going to do now is I'm going to click on the 5HX6P, the prepared protein, and I'm going to duplicate that by pushing right mouse, duplicate, and I am then going to rename the duplicate structure, right mouse, rename, and I'm going to call this chain A. So I'm going to delete that and then chain A. So what I want to do now is to delete the B chains, and that's quite easily done. So you'll notice that I can hover over the different chains like this. So what I'm going to do is I'm going to click on the B chain, and then I'm going to control click on the B waters, and I can then just simply push delete on my keyboard. And that gives me a more simple protein that I'm going to use for docking. So what I'm going to do now is I'm going to show you how we can bring in some molecules in an SD file, how we can dock them in the protein, and what output we get. So I'm going to open a new file. I'm going to bring in some new ligands. So these ligands are very similar to the ones that we already have. And what it's going to do here is I'm just going to push open. I'm just going to accept the defaults we push open. What this will do is it will bring out a new role. So it brought in the new molecules in and put them into a new role in the ligand table which is called the same name as the file, and that has 15 ligands in it. And I'm just going to click on the A chain to select that. And what I'm going to do now is I'm going to, with the 15 molecules selected and my protein selected, I'm now going to go to ligand, and I'm going to go to dock and to go to normal. And what I'm going to do now, so when you set up a docking run, you have the ability to decide how accurate you want your docking to be done. So this will probably depend on how many ligands you want docked. So if this was me in a project and not in a demo, I would probably go for accurate but slow, but normal would probably suffice in most cases. What you'll find, you may want to sort of start with the normal calculation and then see if the results you get are what you were expecting. If not, then you may need to do a slower calculation, like accurate but slow, which might give you some better results. So in this case, I'm going to go for normal. I'm going to define the ligands to dock as the 15 ligands in the ligand table which I've selected. And then I've also selected the protein that I want to dock into. There is a water molecule in the binding site, but I've decided that I'm not going to incorporate that into the docking. So although the water molecule exists, the docking engine will not recognise that it exists unless I click this button here. But I'm going to leave that unclicked. And what I'm going to do is I then also have to define the position that I want the docking to take place in. So in docking, in this particular docking algorithm, it needs to know the position and also in that sort of 3D area, so that 3D volume, it's going to generate an energy grid, so that when it does dock the compounds, it knows how to store them afterwards. So you have to tell the program where the binding site is. So the easiest way, if you've got a ligand, is to select the ligand that defines the sort of in the binding site itself. When you do that, you see just in the background here, you've got you have a blue 3D cube, and so this is what the program is going to use as the binding site for docking. So once you've got the ligand selected, the grid definition selected, and the protein selected, you can then push start to start the calculation. So when you do this for the first time, what happens is that the energy grid is built, and then the docking proceeds after that. Because it takes a while for the docking grid to be calculated, what we do is we calculate it once, and then when you save the Flare project file, that is saved with the Flare project file, so that if in future cases where you want to dock some more compounds at a later stage, you just simply go straight to it. The program will just simply use the existing grid and then dock the compounds using that existing grid. Let me just cancel that. I'm bringing up a Flare project file which has been completed in that respect. Okay, so what I have here then is what you get once that calculation has completed. And if I just scroll up the table a little bit, what you'll notice is that in this in the ligand table now, in this subsection, you have 30 molecules and not 15 any longer, because those more eagle-eyed of you will notice that we were putting some copies of ligands when we were docking. So it keeps the original ligands intact and puts the dock solutions as a copy with a suffix _D in the name. So anything with an _D is a dock solution. The original molecules are still in the ligand table. And so what we can do now actually, if I just want to look at my dock solutions, one thing you can do is on my right hand side here we have another window. So this is service, but we're going to go look at filters. And what I can do with the filters window is I can define it so that I only see the dock solutions in the ligand table. So what I can do then is select title from the pull down, and what I'm going to do now is in this box here I can select containing, And then, because I just want to look at the doc solutions, I simply put _D using my keyboard. And what you can see now is that having put that in there as a filter, what I can see is only 15 molecules. These are the 15 doc solutions, with the other 15 molecules still there, but they've been hidden by the filters. And if I want to see everything again, I can just simply click on this green button here, just to temporarily stop the filter, or simply reapply that to reapply the filter. And what I can show you now is that each of these, so what you're seeing is the doc solutions. And one thing I forgot to mention a little bit earlier, if I just turn this off again, is that these checkboxes here represent, basically, once those are checked, basically the molecule that has been checked stays on all the time. So it means that if you've got a lot of docking solutions like we have here, we can go through the docking solutions and compare it with the crystal structure ligands, so that we have an understanding of how the dock solutions compare to the ligand in the crystal structure. Let me reapply the filter here again. So, of course, what we're seeing at the moment is all the dock solutions have been selected, and so we're seeing them also. What we're seeing, we've got them all of them in the 3D window here. But if I just select one of them, you can see, so the pink one is the original crystal structure, and the sort of the green one is the first molecule in the list. And I should just mention that the colours of the molecule are reflected by the background colour in the ligand view here. So this dark green colour refers to the docked molecule. If I just undo the filter, you see that the crystal structure background is pink, so that reflects the pink carbon atoms in the 3D view. So I've done some dockings. This is the results I get. And actually, you'll notice that there's a little arrow here. These are little buttons. And you can open up the arrows. Then what this reveals is the dock solutions that have been returned once the docking attempt has been returned for each of the compounds. And for each of these, if I give myself a bit more space on the ligand table. So what I'm doing here is doing left mouse button and then dragging the ligand table across a little bit more, so that I have a bit more room. So for each of these docking solutions, you have a number of docking scores that are returned. So you have 3 docking scores that are returned. The one which we kind of use a lot for looking at comparing how well the dockings have done is the LF rank score, and this basically, so in fact all 3 cases are done on a sort of negative scale. So the more negative the number, the better the docking. And by default, the dockings are ranked by LF rank score, going from best to worst. And you can scroll through these by simply clicking on the mouse button and on each of these subsequent solutions, or you can use your up and down arrow keys on your keyboard, like so, and so you can view how your different docking solutions look like. Now it looks like in this case, for the first docking, for this particular case, the first solution looks like it would be the best solution. Sometimes what you find with docking is that because the scoring is quite approximate, sometimes you get some docking solutions which are very similar in their values. So sometimes you might get one docking solution which doesn't look very good, and it'd be the next one down which looks like it's the preferred, it looks like a better pose. And to select that one, all you'll need to do, so let's just for example, if you just want, let's for an example, say that this is the pose that I want, you just simply click on the radio button here, and that defines that as the preferred pose for that particular molecule. But in this case, it's actually I prefer the first one, so I'm just going to click on that. By default, it would be the first pose in the list, which is defined as the preferred pose. What I want to show you now is how we can use the electrostatic, how we can sort of, look at the electrostatic between the ligand and protein, and show you how in Flare we can, you basically have, you can take a set of your aligned ligands and define the protein which they are associated with, and then calculate the electrostatic complementarity for that, for a particular protein. So let me do that now. So what I'm going to do, first of all, is you'll notice that in the columns, before I go any further, you notice that in the column, there's a column in the ligand table called protein. So every time you do something associated with a particular protein, the docked solutions here, because they were docked into this protein, they will be automatically connected to that particular protein, allied to that particular protein. But if you bring in a set of ligands that are aligned in the appropriate way, you bring in some ligands and then align them, they will not automatically align to a protein. So what I want to show you is before we can calculate the electrostatic complementarity, we need to assign the protein to ligands that don't have that assignment. So the pre-docked molecules were actually alignments, and what I'm going to do is I'm going to change my filter now to rather than look at the docked solutions, I want to look at the undocked solutions. So I'm going to look for not containing _D. So we're now looking at the 15 input molecules, if you like, that we've put into the docking. And you'll notice that in this role here, with the 15 undocked solutions, they have no protein associated with them. So what we need to do is select these molecules and assign a protein. So the best way to do this is if you click on this particular button here, so if you click on this button here, this selects all the molecules which are visible in this this role, and so basically these 15 molecules are now selected. And it means that what I need to do now is select the protein that I want associated with these molecules. So to do that, I do right mouse click, and then on one of the 2D structures that have been selected, and then go to set protein, and that will bring up a pull down menu, and you select from that the protein that you want to have associated with these ligands. So we can just put that. So, and you'll notice that in the ligand table and in the protein column, you'll notice that now these ligands have been assigned this particular protein. And this now allows us to calculate the electrostatic complementarity between these ligands and the protein structure.
Yeah.
And so, to do this, now we simply now go to, in the ligand table, we go to the molecular service pull down, and we select electrostatic complementarity.
Sample 66.
So what that is doing now is that it's now calculating the electrostatic complementarity between the proteins that the protein that we've just defined as the associated protein and each of the 15 ligands. So another view that you have, so at the moment what you're seeing is that the 15 ligands are in the same reference frame, and so what we can do actually is if you go to the View tab and Grid View, what you can see is is all the ligands. You can see that all the ligands have now been the electrostatic complementarity. All the ligands have been calculated against that particular protein. And what I'm going to do now is I want to select two particular ligands that I want to compare. Before I do, I've just noticed also that the original ligand, the crystal structure ligand is selected, so I'm just going to uncheck that. And I'm also, what I want to do now is just compare ligand 1 and control click ligand 4. And so what I'm doing here is I want to show you some differences between the two molecules. So I'm going to just control click the protein just to turn that off, because I just want to concentrate on the differences in the complementarity between the two ligands. So compare the complementarity, the two ligands between them and the protein. And so what I'm going to do is, so on the left-hand side, we have a ligand which is micromolar, and on the right-hand side we have a ligand which is nanomolar. And what you'll notice here is specifically in this particular region just here, so I'm just using my left-hand mouse button to twist this round. You'll notice here there's an electrostatic clash between the ligand 1 and the protein, but if I compare ligand 4 and the protein, that clash is all but gone. So very often what you might do and say is, what do I have in that particular part of the protein? Do I have positive electrostatics or negative electrostatics? And so what you can do is, if you go to the ligand tab, you can add the field surfaces, the Z field surfaces that we were talking about a little bit earlier. So we can put the positive on and the negative on. And you'll notice here that on the really active compound, we have sort of a lot of sort of electronegative region in this particular part of the molecule here. In the part of the molecule which is less active, you'll see that yes, there's some electronegativity here, but there's also some positive electrostatics here. And it might be this that is causing some of your detriment to your activity. Another thing that's worth mentioning, you'll notice if I just turn off the positive field just for the moment, you'll notice that there's another section just on the other side of the molecule just here that is looking like a clash. Let me just bring in the protein so that I can show you roughly what might be happening here. So this is the aspartate residue which we had highlighted a little bit earlier. And if I show, so this is the NH, so this is, we're going to be sort of more positive aspect to the region than in where that NH is. And so if I bring back the positive fields again, you'll notice that for the molecule which is highly active, this aspartate NH is in that sort of in that region where there is sort of negativity in the ligand, whereas in this particular ligand, it looks like there's some sort of positive electric field, this is the red region, quite close to this NH. So that may also be contributing to some unfavourable electrostatic interactions between the ligand and the protein. So that is looking at electrostatic complementarity from the surface point of view. The other thing that you can do is generate a score, electrostatic complementarity score for your ligands. So the way to do this is if we use this button to select your 15 ligands again, and then then what I can do is in the ligand tab, calculate EC score. I should just reiterate what you're seeing with the electrostatic complementarity surfaces. Where you have a green region, you have good complementarity between your ligand and your protein. So you have positive electrostatics in your ligand and negative electrostatics in your protein, or vice versa. And red represents an absolute, like a class. So you've got positive region in your ligand, positive region in your protein, and so you've got the spectrum between red and green. So that's what you're seeing as the surface. And as I mentioned in the presentation, you can use that to help you guide you through your sort of, molecule designs. But the other thing that you can do is generate scores for each of the ligands here. If I just scroll across the ligand table here, these have been added to the table at the end here. So we have 3 different electrostatic complementarity scores that are generated. The EC score is the one that is used for the generation of the surfaces, and basically what you're seeing is just a single number here. So what it's doing is it's taking all the vertex points on the surface of the protein and giving you an average score of the ligand as a whole against the protein. So the values that you get for these scores range from +1 to -1, where +1 represents perfect electrostatic complementarity of the protein, to -1 being, if there is such a thing, an absolute clash. So kind of what you want to look for in a lot of projects is to try and maximize this value up towards as close to 1 as possible. But there are different ways you can calculate electrostatic complementarity. So the EC score takes into effect some desolvation. So if there's a desolvation component to the requirements to your protein, this might be a good score to consider. But in terms of if part of your binding event is actually dependent on the electrostatics, then this is the Pearson and Spearman versions of the electrostatic complementarity, and what we find is that these are slightly different scores to this one. And what this represents is these give good correlation with activity if your activity is dependent on the electrostatics. And you can test this out. So what I've got in this data set is the activities already imported into Flare. And what I'm going to do now is in the ligand table, I can go to plot. And what I can do here is I define what I want to see on the x-axis and what I want to see on the y-axis. And so what I will first of all do is select one of the EC numbers, and then on the y-axis define the IC50, the PIC50 on the y-axis, and so this gives you the R-squared value between the EC, the electrostatic complementarity, and the IC50 value. So in this case, there is some dependency on the electrostatic between the activity and the calculated electrostatic complementarity and the activity. And what you can do once you've got this graph up, you can actually also highlight specific residues, so specific ligands, specific points. And what that will do is that will highlight those particular points. These 2 particular points are then highlighted in the main Flare window as well. So you have this ability to generate a plot and look at several select several molecules in that plot, and then that selection will be reflected back into the main ligand table here. So you notice that these 2 molecules these 2 selections refer to design 1 and molecule 13 here. So in a nutshell, that's how you can use electrostatic complementarity to gain a bit more information on your ligand, how perhaps you want to improve your designs on your ligands by improving the complementarity, and actually how you may want to compare how your electrostatic complementarity relates to your known activity. So I could go on for a little bit longer. I've noticed that I've got about 8 minutes left, so perhaps I shall open this up to questions and be happy to take anything that anyone would like to ask. Hello.
Thank you, Stewart, for the presentation. I would like to have one more question based on the dynamics again. Like if we go for a membrane protein simulations, can we have the option of designing the ratio of the lipids within the membrane in the flare?
Could you just repeat your question there? So, I'm going to try and increase my volume on my computer so I can hear you a little bit more clearly. So just bear with me.
I'm just asking that does flare provide the option of designing or building the membrane? Like we have different lipopolysaccharides, and can we have the option of making our own membrane for the simulation in membrane protein simulations in flare?
So are we? So are you talking about like a GPCR, for example?
Okay, I'll write in a chat box. Hello?
So thank you for your question. So at the moment, I'm looking at proteins that are not involved with membranes. So you're looking at, if you like, water-soluble proteins that are not part of the GPCRs. We are looking at at the moment all the proteins are modeled using the amber force field, and at the moment we are trying to implement a new certainly in the future version of Flare, the ability to look at membrane proteins, but at the moment we can't do that.
Okay, thank you.
Sir, one more question. Sir, as you design new ligand on the same core structure, if it is different ligands, suppose we choose 50 different ligands and I want to calculate...
excuse me, your voice is not audible. Can you please speak a bit closer to the mic?
Okay.
Or add your question to the chat, and then I can answer it from there.
Okay.
That's helpful. Can we have an option for target identification if we have synthesized compounds? So I want to, so the next question, I want to calculate EC for different ligand sets. Is it possible? Absolutely. So what you need to do is, for that situation, you'll need to make sure that your ligands are in the same frame of reference as your protein. So what you'll need to do is, so one thing you can do is dock those ligands into the protein, and then do an EC score after you've done the dockings. Or if you're happy, if your ligands are very similar to one another, what you can do is a ligand alignment, so that if your ligands, so if you have a ligand already bound to the protein, you can align your very similar ligands onto your crystal structure ligand, and then calculate the electrostatic complementarity from that. So there are a number of ways you can do it for different ligands, for different ligand series. And then what you can do in what I've shown you just a few moments ago is basically have your different ligand series in different roles. So you could have, in your ligand table here, you could have ligand series 1, and then you have another role, ligand series 2, ligand series 3, and then each of those series would then be assigned, you could assign them slightly different proteins, for example, and check and do the electrostatic complementarity against the protein that you've selected. And in fact, I'm just thinking on the top of my head, the other thing that you can do is maybe you have slightly different proteins that you have in FLAIR, but you want to look at the same ligands in those different proteins. So you align and superimpose the proteins into the same coordinate frame. Then you can then score each of those. You can copy the ligands to a new role, and then assign those copies to a different protein, and then you can start to compare how those ligands look electrostatically with the different proteins as well. So that's something else you could do.
Okay, thank you, sir. Thank you. Please continue.
Stewart, there is another question from Anita Kumari. Can we have option to target identification if we have the synthesized compounds?
Yeah, I was looking at that. That's an interesting question. So I guess the I just wanted to clarify the question. Presumably, what we have is we have some compounds, but we don't know the structure. We don't know the protein which they are binding to. So in that case, what you would do is, say you have maybe 10 ligands that you want, but you don't know the target. So maybe you have target A, target B, target C, target D. So as long as you've got crystal structures for target A, target B, target C, target D, what you can do is you can bring in those 10 ligands, do the dockings against target A, then do the dockings against target B, and then do that for each of the targets. And then you can then start to look at the results and look at so which ligands will best fit which target then. If your targets are very similar to one another, what you can do is do this ensemble docking, which I talked about a little bit earlier, where we align and superimpose all the protein structures into the same coordinate frame, and allow FLAIR to decide which protein it's used in the docking for the different ligands. And when you get the ligand results, when you get the docking results back, the docking solutions are given are allied to the protein which has been docked into. So if you, again, my example of target 1, 2, 3, and 4, if the ensemble docking can basically dock ligand 1 into target 1, it will show that in the ligand table under the protein column, and then the next docking solution might be from target 2, which might be a lower score. So you can make sure that what you have to do is make sure that all the targets are aligned in the same coordinate frame, and then dock your ligands in the ensemble docking mode.
Can you explain a bit about radial plots?
radial plots. So yeah, so I'm thank you, Dr. Dinesh, for that question. So so basically, radial plots are basically a way of taking lots of different values and providing a sort of a consensus score, if you like, a global score for all that involves all those those individual scores. So you could have a score for the electrostatic complementarity. You can have a score for docking. In fact, we could try it now, and let me just see if we can. It won't take too long to do. So let me see if I can bring something up and show you. So what we have here is our 15 molecules, and what I'm going to show you here on the right hand side, I'm going to bring up the radial properties window. So do that view docs, and then go to radial properties, and then what you can do here is you can select any numerical values that you have in the ligand table to be part of the radial property. So what we can do here, I'm just going to start from scratch actually. We could define, for example, the EC row score. So here you say that the value below 0.2 is a poor score, the value above 0.4 is regarded as a good score, and anything in between is an intermediate score from poor to good. And you can do the same for let's try something. So let me just try. Let's find the PIC50 here. Where's that? That should do it. So again, the better score is for PIC50 is obviously above 8 is getting up to nanomolar, below 6 is below micromolar, and then you can then also have perhaps the docking score. So you notice that as I add more functions to the radial properties, the shape changes, and so what this ends up being is like an overall score from the individual score. So if you've got lots of different competing features that you want to take into account when you're selecting your molecules, using the radial properties allows you to take into all these different features and make one single score out of them, so that you can rank on that particular score.
Hello, sir.
Hello.
So in the graph versus EC50 and the PIC50 values, the ligand file which we have to introduce in the flare should have the known IC50 values, only then we can get that graph, na?
Yeah, that's right. So in this example, so what I would do if I was starting a new project, what I would do is download the known molecules from Chemble, so which has the IC50 values already sort of stored in the data set. So you have to, or if you have a biology team that have given you the IC50 values, then these have to be in the SD file which you bring into Flare. Or it could be a CSV file. So what you can do is you can open a particular CSV file, and you go through a series of steps where you import, you define which of your columns refers to your structure, and then hopefully that will bring in the structure, and then also will add the columns including your activity if the biologists have given you that.
Okay, sir. Thank you.
Thank you very much.
Yeah, please hold on. There is another question.
Sir, I have a question, but it is not from Flair. It is from scaffold hopping. Actually, I want to know if I have 4 substitution group in a ligand. I want to modify 4 places at the same time.
You're gonna mute.
So your voice is not audible. Can you type?
You're on mute. Yeah.
Okay.
So I've just got the chat from the chat. I have an R group substitution in a ligand. I want to modify R1, R2, R3, and R4 position at the same time. Is it possible? So how's best to answer this now? So, yeah, so I think what my answer will be this in the following actually. So what you need, what Flair doesn't do is at the moment automatically add R groups to your system. What you have to, at the moment you can manually do that with the editing tool that I showed you. So of course you can do that now. But in a future version of Flair, you should be able to, if not do it at the same time, doing it at a very similar time, to add the R1, R2, R3, and R4 groups to your core molecule. And that in terms of a library enumeration tool. This is not available currently in Flair 5, but we're hoping that it's going to be available in a future version of Flair. Oh, in Spark module. Okay, sorry. So in Spark, you define one. Let me just think about that for a second. Just think about that for a second. That's a good question. I haven't tried it myself, and you definitely, what you may need to do is do it one at a time. Let me just think about that. No, so you have to basically do that one at a time, so that so you'll do one R group, and then perhaps bring the solutions into Flair, select the ones that you think are that look good, and then look at R2, then R3, then R4. So at the moment, you do one R group at a time.
Yeah, thanks Stuart for your answer to the queries, and I'm sure that there will be many more questions, and people can send us email or directly email to you for further questions.
I'll be very happy to help out, Dr. Dinesh, and thank you so much for your time today and for allowing us to talk. I really, really appreciate it. Thank you so much.
Yeah, thank you very much for joining.
Thank you.
Thank you. Bye.
Thank you. Thank you, Dr. Dinesh, for the opportunity.
Thank you. Welcome.
No problem.
Thank you, Dr. Dinesh.